In the soft darkness of night, when the world around you has quieted into a slower rhythm, it becomes easier to imagine the landscapes hidden within your own body, places so small and yet so complete that they resemble tiny worlds of their own. As you settle into a comfortable position, the boundaries of your awareness can gently drift inward, moving slowly from the familiar surface of the skin, through layers of tissue, and deeper still, until your focus seems to rest inside a single living cell. It is not a hurried transition, but an unhurried descent, guided by the same calm steadiness that governs all of biology. Inside this imagined space, everything seems to move with a deliberate softness, as though each molecule understands its role and carries it out with quiet patience. Nothing feels rushed here. Nothing jostles for attention. Instead, this microscopic world unfolds with the same gentle logic that organizes the body as a whole, one that maintains harmony through balance, repetition, and the subtle collaboration of countless tiny components. The cell you've entered might be any one of the trillions that form your body, each performing its duties in coordination with others, all contributing to the stability of life. Much like how organ systems work together to maintain homeostasis, regulating water levels, temperature, calcium balance, and other delicate conditions throughout the body. This tiny cell maintains its own internal equilibrium with remarkable accuracy. The environment you find here feels warm, stable, and enclosed, almost like drifting inside a peaceful room tended by invisible caretakers. Though the cell is far too small to perceive with ordinary senses, the imagination can conjure a sense of being buoyant within a clear, gently illuminated space filled with soft currents. Around you is the cytoplasm, a calm, gel-like sea where proteins, ions, and organelles drift in unhurried patterns. This fluid isn't still, but moves with a slow motion reminiscent of a quiet tide. It carries nutrients, signals, and fragments of energy, all directed by subtle chemical gradients rather than any obvious wind or push. Each movement is governed by the principles of chemistry and physics, the same foundational laws that guide ecosystems, weather patterns, and the grand machinery of nature. But at this scale, those laws feel intimate and quiet, revealing themselves through soft interactions rather than dramatic force. Suspended within this cytoplasmic landscape are structures that would be striking if they were large enough to see. The mitochondria, with their soft folds, reminiscent of gentle waves, the endoplasmic reticulum spreading like serene channels, and the Golgi apparatus, stacked in calm layers. Yet your attention is drawn toward the center of the cell, where the nucleus rests like a quiet sanctuary. Its membrane, smooth and protective, encloses the cell's genetic library. The nucleus is a place of order within a place of motion, a gentle anchor in the constant ebb and flow of cellular activity. The membrane itself is soft but purposeful, dotted with nuclear pores that act as guarded gateways. Through these openings, molecules travel in and out, some carrying messages, others bringing materials for copying, repairing, or adjusting the DNA stored inside. These movements are not chaotic. Each passage is controlled and selective, so the nucleus maintains an environment as carefully balanced as the broader body does when adjusting to temperature or regulating minerals. Everything in biology, from the scale of organs to the scale of molecules, depends on this same principle of regulated flow, where systems respond to change with quiet correction and subtle coordination. As you drift closer to the nucleus in this imagined space, its subtle glow becomes a focal point. Though DNA itself does not emit light, thinking of it as a soft, luminescent coil can help the mind relax into the metaphor of life's memory, resting here, protected and cherished. The nucleus holds the entire blueprint for the organism, written in long, spiraling molecules that encode instructions for every structure, every protein, and every process that keeps the cell alive. This DNA is not rigid. It floats in its own gentle environment, wrapped around histone proteins, coiling and uncoiling as needed in patient rhythmic motions. The double helix itself feels serene, its two strands running side by side like quiet partners, connected by pairs of nucleotides that resemble tiny steps on a twisting staircase. These base pairs, adenine with thymine, cytosine with guanine, hold together through weak but persistent attractions, forming a structure that is both stable and flexible. This balance between steadiness and adaptability is essential. It mirrors the balance found throughout the body, where each organ system contributes to maintaining a stable internal environment while adjusting to external changes. The cell, like the entire organism, thrives on this interplay between consistency and responsiveness. Although the DNA appears peaceful, it is far from static. While you rest, trillions of these actions unfold throughout your body in perfect silence, without any conscious effort or sensation. The DNA is constantly communicating instructions for producing proteins, guiding the cell through its daily tasks with steady precision. And even when nothing seems to be happening, repair systems quietly monitor the strands, looking for small irregularities or signs of wear, much like how the body as a whole monitors for fluctuations in temperature, fluid levels, and nutrients to preserve stability. Inside the nucleus, this subtle maintenance ensures that the DNA remains reliable across time. It is almost soothing to consider the sheer scale of this process. Every day, each cell experiences countless minor disturbances, tiny chemical changes, shifts in the structure of a nucleotide, or small imperfections introduced when DNA is copied. Yet these disturbances are addressed calmly and promptly. Repair enzymes move across the DNA as though gliding, inspecting each region and responding to even the most minimal irregularities. Their work is not dramatic. It resembles a soft, unending form of housekeeping. This first moment within the cell is meant to settle the mind into recognizing the extraordinary order that exists beneath the surface of life. Just as the broader body uses interconnected systems to maintain homeostasis, every cell uses quieter but equally sophisticated systems to protect its DNA. Water balance, temperature regulation, glucose monitoring, and calcium adjustments at the organismal level parallel the constant balancing acts within the cell, where conditions must be ideal for the DNA to rest and function. As you imagine drifting deeper into this environment, the peacefulness of the nucleus begins to feel like a metaphor for the body's resilience. The DNA, though delicate in appearance, is supported by layers of stable structure, careful monitoring, and gentle correction. Nothing within the cell expects perfection. Instead, the system is designed for quiet repair, slow adjustments, and continuous renewal. Even though damage happens simply through the act of living, the cell is equipped with a sophisticated repertoire of responses that restore balance again and again. The descent into the cell ends on this note of calm continuity. All around you, imagined or real life, sustains itself through gentle cycles of preservation and repair. The nucleus with its softly coiled memory, the cytoplasm with its unhurried currents, and the organelles with their measured rhythms all contribute to a sense of quiet harmony. Even at this microscopic scale, the principles that guide the body remain the same: cooperation, balance, and a steady return to equilibrium. And as the scene settles into stillness, you carry with you the awareness that every moment, even now, your cells are performing these quiet acts of care, maintaining the blueprint of life with a resilience that never sleeps. 2. DNA as the silent memory of life. In the quiet interior of the cell, the nucleus rests like a softly glowing chamber, and within it lies the molecule that has shaped every moment of your existence. DNA, the silent memory of life, stretches out in long, elegant strands that hold the gentle instructions guiding each cell's behavior. To imagine this molecule is to imagine a kind of internal calm, a blueprint written with extraordinary precision, yet stored in a form so delicate that it seems almost poetic. As you ease further into this reflective space, the presence of DNA begins to feel like a soft current of continuity, extending backwards to your earliest beginnings and quietly forward into every moment yet to come. If you picture the double helix, perhaps it appears first as a smooth ribbon twisting slowly through a tranquil environment. Each curve feels unhurried, each rotation steady and purposeful. The structure is often shown as two spiraling lines, but in this imagined view, it is easier to sense it as a pair of gentle streams flowing side by side, connected by thousands upon thousands of tiny bridges. These bridges are the base pairs adenine with thymine, cytosine with guanine, linked by bonds that are strong enough to hold the strands together, but soft enough to be separated when the cell needs to read or copy the genetic message. This balance between stability and flexibility is essential, echoing the way the entire body balances itself through constant, subtle adjustments to maintain homeostasis, regulating everything from temperature to water levels to mineral concentrations in the blood. The DNA molecule is composed of three simple components: a sugar, a phosphate group, and one of four nitrogenous bases. Although these components are tiny and uncomplicated on their own, their arrangement within the double helix creates an astonishingly efficient storage system. Imagine a library where each book contains detailed instructions for constructing a specific protein, and every book is written using only 4 letters. The simplicity of this alphabet is almost soothing. Despite having only 4 types of bases, the order in which they appear along the strand forms an intricate code that shapes the entire organism. Each sequence is a quiet directive, a softly spoken phrase in the language of life, even though DNA carries immense quantities of information. This is possible because the molecule is carefully wrapped around histone proteins, forming a structure called chromatin. If the DNA were fully stretched out, it would reach lengths far greater than the size of the cell itself. Yet through this graceful packaging, it rests comfortably within the nucleus, coiled and folded with elegant efficiency. Thinking of these coils as gentle loops or flowing strands can help the mind picture how DNA remains both compact and accessible. The chromatin fibers can loosen when needed, allowing repair enzymes or transcription machinery to reach specific regions before returning to a more restful, compact state. This dynamic, shifting organization might seem busy, but in truth it moves with a slow and unhurried rhythm. The cell adjusts chromatin structure gradually, responding to signals with a kind of patient attentiveness. When a gene needs to be read, small sections gently unwind, exposing the bases so that the cell's machinery can copy their sequence into RNA. When repair is needed, the chromatin opens just enough to let enzymes slip in and restore the integrity of the DNA. This constant, quiet choreography reflects the cell's commitment to preserving the genetic information with care, much like how the broader body protects equilibrium through countless subtle interactions among the organ systems. The idea that DNA serves as a memory is soothing in itself. Every segment of this molecule carries echoes of ancestral life, shaped by countless generations before you. Evolution, as described in the biological literature, is guided by descent with modification, a gradual shift in heritable traits passed from one generation to the next, while still preserving the essential unity shared by all living organisms. DNA is the medium through which this continuity is maintained, holding within its sequence the accumulated experiences of life across deep time. Even as species diverge and adapt, the underlying structure of DNA remains constant, a shared thread linking life across billions of years. Within your own cells, the DNA is not merely a passive archive, but a living document. Its instructions are read continuously, not to produce anything dramatic, but to maintain the calm rhythm of cellular function. Proteins are synthesized, membranes replenished, enzymes produced, and signals conveyed, all based on the information resting in the double helix. The process is so steady and dependable that it becomes easy to forget the astonishing accuracy required. Every second, countless cells within your body rely on DNA to provide precise directions so they can perform their roles. Whether those roles involve carrying oxygen, transmitting neural signals, contracting muscle fibers, or maintaining the structure of tissues. As you imagine the DNA drifting peacefully inside the nucleus, it becomes easier to appreciate how perfectly adapted it is to its function. The opposing strands allow for redundancy. If one is damaged, the other can guide repair. The base pairing keeps the structure stable while still allowing it to open when necessary. The phosphate backbone provides strength, like the rails of a ladder holding everything in place, and the sequence of bases, stretching along the molecule in long, unbroken patterns, forms a quiet narrative describing every aspect of cellular behavior. Inside this gentle environment, the DNA rarely remains fully exposed. It is shielded within the nucleus, protected by the nuclear envelope and guarded by a team of proteins that monitor its condition. The nucleus acts as a peaceful chamber, isolating the genetic material from the constant activity of the cytoplasm. This separation preserves the calm environment needed for accurate reading and copying. Only carefully selected molecules pass through the nuclear pores, maintaining order and preventing disruptions. Even when the cell prepares to divide, the DNA condenses further into tightly packed chromosomes, as though wrapping itself in protective layers before undergoing the complex choreography of replication and separation. In the wider context of the body, this organization reflects a theme found throughout biological systems: the careful layering of protection around essential functions. Just as the brain is shielded by the skull, membranes, and cerebrospinal fluid, the DNA is shielded within the nucleus, insulated from unnecessary disturbance. Layers of structure, from the atomic composition of the nucleotides to the organelle-level arrangement of chromatin and nucleus, demonstrate the hierarchical organization that characterizes life at every scale. This hierarchy, from molecules to cells, from tissues to organs, and from organs to interconnected body systems, ensures that each part contributes to the stability of the whole, all working together in quiet collaboration, as described in the broader literature on biological organization. As this imagined scene continues to unfold, the DNA feels less like a complex molecule and more like a serene presence. It is a stable thread of meaning woven through the entire fabric of the organism. Every cell carries a copy of this thread, and though only certain parts are used at any moment, the full archive remains intact, resting quietly in the background. There is something reassuring in knowing that such a gentle but powerful system resides within you. The DNA does not seek attention. It does not create noise or turbulence. Instead, it ensures that life proceeds with order, coherence. Allowing your awareness to rest here, in the company of the double helix, you may feel a growing sense of ease. Just as the DNA maintains the instructions for your physical existence, it also symbolizes continuity, resilience, and balance. Through countless cycles of change, cell division, repair, growth, adaptation, the DNA endures with steady poise. Its structure supports flexibility without chaos, change without disorder, and a kind of internal memory that quietly shapes the body throughout life. In this moment, the DNA remains suspended in its soft environment, cradled within the nucleus, surrounded by the gentle ebb and flow of cytoplasmic currents. Its presence is both simple and profound, a quiet testament to the deep harmony that underlies the biology of every living being. And as you drift deeper into rest, the silent memory of life continues its calm vigil, preserved within every cell, guiding each moment with patient precision. 3. The double helix, a harmonious structure. When you imagine yourself drifting once more into the quiet interior of the cell, where the soft glow of the nucleus spreads through an otherwise tranquil landscape, the double helix begins to appear again, rising slowly out of the stillness like a ribbon suspended in water. Its two strands curve in a gentle spiral, not hurried, not tense, but resting in a state of perfect poise. The harmony of its structure is something you can feel even without seeing it, a peaceful order that helps anchor the cell's internal world. The double helix is not merely a shape. It is a living embodiment of stability and balance, formed through the repeated pattern of molecular interactions that echo the broader principles of biological organization found throughout the body. As the helix rotates in your mind's eye, its beauty comes from its consistency. Each turn of the spiral follows the same rhythm, winding around an invisible axis that holds the structure together with delicate strength. The strands themselves, made of alternating sugars and phosphate groups, form a kind of gentle backbone. This backbone does not demand attention, yet it supports everything else. It is the same kind of understated support that exists throughout the body, like the silent work of the bones holding up the skeleton, or the steady beat of the heart quietly circulating blood long before you ever think about it. In the body, stability often happens in the background, and the double helix follows this pattern with elegant restraint. Between the two strands, the bases pair in a soft, repeating sequence. Adenine pairs with thymine, cytosine with guanine, each connection formed by hydrogen bonds that feel almost like gentle touches, a kind of molecular handshake that keeps the two strands aligned. These bonds are not rigid. Instead, they exist with just the right amount of strength and subtlety, allowing the strands to separate when needed for replication or repair. They are stable enough to maintain the code of life, yet flexible enough to allow the entire molecule to breathe and move. This gentle balance between strength and pliability is crucial, for if the structure were too rigid, it would resist the motions essential for reading or copying genetic information, and if it were too loose, the instructions written within it could drift apart or degrade. The bases themselves stack like quiet pillars, their edges forming a smooth interior surface. This stacking creates a sense of calm uniformity, with each rung of the helix contributing to the overall stability of the molecule. When you think of the bases arranged this way, in orderly columns that rise like the steps of an ancient spiral staircase, the image becomes almost meditative. It suggests that even within the complexity of life's instructions, there is room for simplicity and order. The double helix, with all its biological importance, is fundamentally a repeating pattern, not unlike the repeating rhythms of breathing, heartbeat, or steady footsteps walking across a quiet landscape. Because DNA is so long, stretching across millions or even billions of base pairs depending on the organism, its structure must remain both manageable and efficient. The harmonious shape of the helix helps accomplish this. It ensures that the molecule does not form chaotic tangles or knots, but instead coils smoothly, folding into chromatin and eventually into chromosomes when the cell prepares to divide. This higher-order packaging reflects the hierarchical nature of biology itself, where small structures combine to form larger ones, each level supporting the next. Just as atoms form molecules, molecules form organelles, and organelles form cells. The repeating units of DNA come together seamlessly to create a coherent whole. The elegance of the structure lies not only in its appearance, but in its ability to support life at multiple levels of organization. As you imagine the double helix in the quiet nucleus, you may sense how the environment around it contributes to its serenity. The nucleus creates a protected space where the DNA can rest without interference from the constant activity outside. The nuclear envelope shields it with layers of membrane, and the chromatin structure cushions it with graceful loops and folds. It is almost as if the cell recognizes the importance of this molecule and has created a sanctuary where it can exist free from unnecessary turbulence. This sense of shelter echoes the body's broader tendency to protect what is essential, or regulated internal environments that keep systems functioning smoothly even as external conditions shift. The shape of the double helix also plays a central role in how the cell reads and interprets the genetic code. When the strands need to unwind so that a specific gene can be transcribed into RNA, the helix responds with a slow and deliberate opening. The bonds between the bases separate one by one, allowing the cellular machinery to glide along a single strand, copying its information into a new form. This process happens quietly, without drama, and in rhythm so steady that it mirrors the gentle cycles of nature, growth, rest, renewal, The harmonious structure of the helix ensures that these cycles occur without strain, allowing the cell to respond to its needs with calm efficiency. The uniformity of the double helix also allows it to be faithfully copied during cell division. When a cell prepares to divide into two daughter cells, each one must receive a complete and accurate copy of the DNA. The helix unwinds, and each strand serves as a template for the formation of a new complementary strand. Because the structure is so regular, the process unfolds with remarkable accuracy, ensuring the continuity of life from one generation of cells to the next. This predictability is essential, echoing the way the body maintains stable internal conditions across time, adjusting to changes while preserving balance. Despite the calm appearance of the helix, it is not immune to disturbance. Small chemical changes, slight shifts in structure, or the influence of external factors can nudge the molecule out of alignment. Yet, even in these moments, the harmonious nature of the helix supports resilience. The structure's clarity allows repair enzymes to recognize deviations from the expected pattern. They move along the DNA with slow purpose, reading the sequence and identifying areas where corrections are needed. The regularity of the helix makes mistakes noticeable, and this visibility ensures that damage can be addressed before it spreads or disrupts the delicate balance of cellular life. Thinking of the double helix as a harmonious structure helps illuminate its deeper significance. It demonstrates that life's complexity rests upon patterns that are fundamentally simple and orderly. The pairing of four bases, the repeating sugar-phosphate backbone, the consistent twist of the helix, these are the building blocks of an entire organism, from the emotional landscape of the brain to the quiet strength of bone. Even the most advanced processes of growth, adaptation, and evolution depend on the steady reliability of this structure, whose form remains conserved across nearly all life on Earth. Evolution has shaped the content of DNA. Its sequences change, adapt, and diversify through countless generations. But the architecture of the double helix remains a unifying feature shared by every species, a gentle reminder of the interconnected history of life. As you let the image of the double helix drift slowly in your imagination, its symmetry and grace can evoke a sense of peaceful alignment. It stands as a quiet symbol of continuity, a structure that carries ancient memory through the present moment and into the future. Every cell that houses this molecule participates in preserving that continuity, and the helix itself, turning endlessly through the silent space of the nucleus, remains a testament to the deep harmony that supports life at every level. In its gentle curves and balanced bonds, the double helix offers a vision of order and calm that reflects the entire biological world, guiding each cell with unwavering, quiet clarity. 4. Everyday sources of DNA wear. As you return once more to the quiet interior of the cell, where the double helix rests in its softly spiraling form, it becomes comforting to remember that even in this peaceful environment, the DNA is constantly experiencing gentle, almost imperceptible disturbances. These sources of wear are not dramatic intrusions or sudden shocks. Instead, they arise naturally from the ordinary rhythms of life, unfolding with the same subtlety as the slow drift of cytoplasmic currents or the steady beating of the heart in the wider body. In a sense, the DNA lives within a world where small fluctuations are simply part of existence, much like how the body continuously responds to changes in temperature, water levels, or nutrient availability to maintain homeostasis. Nothing remains perfectly still, and nothing remains entirely untouched. Yet this ongoing, quiet exposure to change is met with the cell's remarkable ability to correct, restore, and continue forward with gentle resilience. One of the most common sources of DNA wear arises from the chemistry of life itself. Within the cell, countless reactions occur every second, generating energy, building molecules, and breaking others down. These reactions form the foundation of metabolism, allowing the cell to grow, respond, and function. But in the process, they also produce byproducts that can subtly influence the DNA, Reactive oxygen species, for instance, appear naturally as part of energy production in the mitochondria. These small, reactive molecules drift through the cytoplasm and can occasionally come into contact with the DNA, altering a base or shifting a bond. The effect is usually slight, almost like the faintest brush of wind against the surface of a still pond. Yet even these delicate interactions can change the structure of a nucleotide, creating the need for gentle correction. Temperature, too, plays a role in the gradual wear on DNA. Though the body maintains a remarkably stable internal temperature through coordinated actions between the cardiovascular, muscular, integumentary, and nervous systems, slight variations still occur throughout the day. The warmth generated by movement, the coolness of rest, the subtle changes influenced by environment all contribute to tiny fluctuations that influence the chemical stability of the DNA. Heat encourages motion at the molecular level, causing bonds to vibrate more intensely, which can occasionally lead to spontaneous changes in the bases or even cause the two strands of the helix to separate for brief moments. These shifts are so small that you might imagine them like gentle ripples on an otherwise smooth surface. Another source of everyday wear comes from the process of DNA replication itself. Every time a cell prepares to divide, it must copy its entire genetic library, producing two strands that mirror the original. This process is incredibly accurate, unfolding with a precision that might be compared to a skilled artisan replicating a detailed pattern by hand. Yet even the most careful work can contain small imperfections. When the polymerase enzyme travels along the DNA, reading each base and adding its complementary partner, it does so with immense reliability. But occasionally a mismatch slips through. Instead of pairing a cytosine with a guanine, it might mistakenly introduce a different base. These copying errors represent some of the most common and gentle forms of DNA wear, arising not from an external threat but from the very act of perpetuating life. They reflect the same principles seen throughout biology. Even systems designed for precision still operate within the subtle boundaries of natural variation. Chemical instability also contributes to ongoing DNA wear. Certain bases can undergo spontaneous transformations under normal physiological conditions. Cytosine, for example, can gradually lose an amino group in a process called deamination, converting into uracil, a base not typically present in DNA. This kind of subtle change occurs without any external influence, just the quiet workings of molecular chemistry over time. Similarly, the bonds that connect the bases to the sugar-phosphate backbone can occasionally break, creating what is known as an abasic site, where a base is missing entirely. These events occur slowly and infrequently, but given the vast length of DNA, they accumulate with gentle regularity, like soft drops of rain occasionally disturbing a calm surface. Even the cell's environment contributes to DNA wear in ways that feel subtle rather than disruptive. Nutrient levels shift depending on the cell's activity, and changes in pH or ion concentration can influence the stability of the genetic material. The body works constantly to maintain these parameters within a narrow range, coordinating between the digestive system, endocrine system, cardiovascular system, and others to keep the internal environment steady. Yet within this overall stability, small oscillations occur, each one creating a slightly different chemical landscape that the DNA must quietly inhabit. These shifts are not threatening. Instead, they are part of the natural ebb and flow of life, a reminder that the body is always adjusting, always finding its way back to equilibrium. External influences, while often imagined as dramatic sources of DNA damage, also arise through ordinary, gentle interactions with the world. The soft glow of sunlight, something most people experience daily, carries ultraviolet rays that can alter the bonds between adjacent bases, particularly thymine. These rays do not harm the cell immediately. Instead, they cause slight distortions in the DNA's shape, creating small bends or kinks that the repair systems later address. Even the food we eat, the air we breathe, and the natural chemicals we encounter as part of daily life can introduce mild influences on the DNA. These interactions are not inherently harmful. They simply represent the continuous dialogue between the organism and its environment. Physical processes within the cell also contribute to DNA wear. The helix is not rigid. It opens and closes frequently as the cell reads genes, transcribes RNA, or allows repair enzymes to glide along its length. This constant motion introduces opportunities for small strains or shifts in the structure, similar to how repeated bending of a gentle material can eventually create small signs of use. But these motions are necessary, allowing the cell to access the information stored within the helix and ensuring that the instructions for life remain active rather than locked away. The DNA is designed for this flexibility, yet each motion still represents a moment where the molecule adjusts, yielding to the needs of the cell. Through all of these influences, chemical reactions, temperature changes, replication errors, spontaneous shifts, environmental interactions, and the natural mobility of the molecule, the DNA experiences continuous, quiet wear. None of these sources is inherently alarming, and none threatens the stability of life on its own. Instead, they are gentle reminders that biological systems exist in a state of constant motion, never perfectly still, yet always striving for balance. The DNA endures this ongoing activity with remarkable grace, supported by the cell's intricate repair systems that work steadily and silently to correct any changes that arise. As you let your thoughts linger on this idea of everyday wear, it becomes easier to see the double helix not as a fragile structure, but as a resilient one. Its harmony comes not from being untouched, but from being supported by layers of biological care. The molecule continues its patient vigil, carrying life's memory through each small shift, each gentle influence, each natural fluctuation. And in this quiet endurance, the DNA reflects the broader truth of living systems, that life persists not through absence of change, but through the graceful ability to meet change with calm and steady repair. 5. The cell's quiet surveillance network. Inside the serene interior of the cell, where the double helix rests in gentle spirals, and the cytoplasm moves with slow, drifting currents, an intricate and unceasing watchfulness unfolds. This vigilance is not loud or forceful. It does not resemble alarms or warnings that might be found in the larger world. Instead, it resembles a soft, attentive presence, an awareness woven into the fabric of the cell itself. The cell's quiet surveillance network continually observes the state of the DNA, monitoring it with calm precision and responding to even the slightest irregularities with a kind of molecular tenderness. It is here, in these unhurried and almost imperceptible interactions, that the foundations of DNA repair begin. To imagine this surveillance, it helps to picture the nucleus as a tranquil chamber where nothing moves hurriedly. Enzymes glide through this space like gentle caretakers, drifting along the chromatin fibers with unspoken purpose. They do not need urgency to perform their work. Instead, they explore the DNA in a steady, unbroken rhythm, brushing lightly against the double helix as though reading its texture. These proteins are tuned to recognize what the structure should feel like. When a slight variation appears, a base out of place, a bond weakened, a slight bend where none should be, they sense it, the way fingertips might sense a small wrinkle in fabric. The recognition is subtle, a quiet shift that draws their attention, not through panic but through the simple contrast between harmony and irregularity. Within this calm environment, many different proteins serve as guardians. Some move along the DNA in long stretches, scanning for distortions. Others remain positioned at specific locations within the chromatin, ready to respond when nearby damage is sensed. Still others float freely, responding to chemical signals that indicate a disturbance has occurred somewhere in the nucleus. Their movements are not random. They follow gradients, cues, and molecular landmarks that guide them gently to the places where they are needed. Every part of this network reflects the same broader principle observed in the physiology of the whole body: coordinated, continuous regulation that maintains internal balance, despite the small fluctuations that occur naturally over time. Just as the body adjusts water levels, temperature, or blood glucose to preserve homeostasis, the cell adjusts the state of its DNA with patient attention. One of the most remarkable aspects of this surveillance system is that it operates long before damage becomes threatening. Most of the time, the changes it detects are small enough that the DNA's overall structure remains stable. A single base might shift its chemical orientation, or a bond might be temporarily weakened. Even so, the cell's guardians respond with gentle consistency, recognizing that preserving the integrity of the genetic code requires addressing disruptions at the earliest stage. This proactive care is one of the reasons DNA remains so resilient. Rather than waiting for problems to accumulate, the cell maintains a constant awareness that allows it to respond with quiet precision. To visualize the way these proteins sense DNA, you might imagine them drifting along the strands. Each enzyme has a particular shape, formed through the folding of its amino acids, and this shape determines what it can recognize. When it touches a region of DNA, it can feel whether the bases are paired correctly, whether the helix maintains its usual twist, and whether any part of the backbone has been broken or shifted. This interaction is subtle, almost like reading Braille at the molecular level. The proteins do not need to unravel large sections of DNA to understand its condition. They simply sense the physical texture and chemical properties of small, local regions, detecting irregularities with extraordinary sensitivity. This sensitivity extends beyond the structure of the helix. Many surveillance proteins also recognize chemical modifications that appear on the bases, such as oxidation, deamination, or alkylation. These changes are often the result of ordinary metabolic processes, the quiet chemistry of life that unfolds endlessly within every cell. When a base undergoes such a transformation, its properties shift slightly, creating a difference that the cell's guardians can detect. It is a reminder that the DNA exists within a dynamic environment, influenced by the natural activities occurring around it. Yet the surveillance network responds to these influences with the same steady, calm, guiding repair processes to the site of change before the alteration can interfere with the DNA's ability to function. Another part of the cell's quiet surveillance involves tracking the timing and conditions of the cell cycle. The DNA is more vulnerable during replication, when the strands separate and the bases become briefly exposed. During this time, the cell increases its vigilance, bringing more guardians into contact with the DNA and slowing certain processes to allow for more careful monitoring. Checkpoints exist throughout the cell cycle, gently pausing progression if anything appears irregular. These pauses are not abrupt stops, but rather smooth, measured delays that give the cell time to ensure its DNA remains intact before moving forward. In this way, the cell's internal rhythms adapt to the needs of the DNA, adjusting their pace like a musical composition that slows briefly before continuing with renewed harmony. This network of surveillance also communicates through chemical signals, most of them quiet and diffuse. When damage is detected, messenger molecules spread through the nucleus, attracting repair enzymes and orchestrating the coordinated response that will restore the DNA's structure. These signals do not create urgency. Instead, they provide information that gently shifts the balance of activity within the cell. Enzymes that normally perform other tasks move toward the site of damage, while certain processes slow down to allow the repair to unfold without conflict. Everything moves toward the same goal, preserving the integrity of the genetic code. The surveillance system is also capable of distinguishing between different types of wear. A small disruption to a single base is handled differently from a larger distortion that affects multiple nucleotides, and both are treated differently from breaks in one or both strands of the DNA. The guardians recognize these variations, not through conscious choice, but through the ways specific proteins interact with the altered structure. The cell does not need to think about its responses. The correct pathways emerge naturally from the interaction between the proteins and the DNA. This effortless coordination reflects one of the deep truths of biology. That complex behavior often arises from simple interactions repeated across time. Beyond the nucleus, the cytoplasm also participates subtly in this quiet vigilance. Signaling pathways extend between the nucleus and the cytoplasm, allowing the cell to adjust its metabolism or redirect its resources based on the state of the DNA. If the nucleus signals a need for additional repair enzymes, the cytoplasm responds by synthesizing them. If a more extensive form of repair is required, each adjustment is made with the goal of preserving stability, much like the cooperative efforts of organ systems maintain balance throughout the body. As you envision the surveillance network at work, it feels less like a system of defenses and more like a continuous form of care. The DNA is not protected through force or urgency. It is watched over through attention, recognition, and response. The cell has no concept of threat, only the steady awareness of structure and harmony. When that harmony is disrupted, even subtly, the network responds with gentle precision, restoring the DNA to its natural state. In this quiet environment, where the double helix floats in a softly lit chamber, the cell's guardians continue their work without pause. Their movements are patient, their interactions calm, their vigilance unwavering. They ensure that the molecule containing the memory of life remains whole, supported by layers of attentiveness that exist not in flashes of intensity but in the steady flow of molecular awareness. And as you imagine this watchfulness, it becomes easier to appreciate the remarkable resilience of life's blueprint, protected, guided, and restored by the cell's constant, quiet care. 6. Base excision repair, a gentle cleanup crew, within the soft and steady rhythm of the cell, where the double helix rests like a calm spiral and the quiet surveillance network moves patiently across its length. There exists a repair system designed to handle the smallest, most delicate imperfections that naturally arise in DNA. This system, known as base excision repair, works not with force or urgency, but with a kind of molecular tenderness, addressing the subtle changes that happen simply because life is always in motion. These changes are not dramatic breaks or large distortions. They are tiny shifts in the chemical nature of individual bases, little irregularities that occur through the gentle chemistry of living processes. And in response, the cell deploys a quiet cleanup crew enzymes that roam the helix with soft precision, recognizing and correcting these small deviations to preserve the harmony of the genetic code. To imagine this process, you might picture the DNA as a long ribbon drifting inside the tranquil space of the nucleus. Along its length, single bases occasionally lose their clarity, not enough to disrupt the entire strand, but enough to create a small difference that the cell's guardians can detect. A cytosine might undergo deamination, becoming uracil, a base more common in RNA than DNA. An adenine might be slightly oxidized, or a guanine might shift its orientation. These changes are subtle, often the result of metabolic byproducts or natural chemical oscillations occurring within the gentle warmth of the cell. Nothing explodes or tears. Rather, the bases experience small nudges, like plates on a table shifting a fraction of an inch. Yet even these tiny movements require correction, for the genetic code relies on precise pairing to guide the cell's activities. The first responders in this quiet repair system are enzymes known as DNA glycosylases. These proteins travel along the DNA in a smooth, unhurried glide, brushing against each base as though feeling for slight differences in texture. Each glycosylase is tuned to recognize a specific type of damage. One may detect uracil where it should not be, another may sense an oxidized guanine, yet another might look for bases that have been improperly methylated. They do not hurry. Instead, they move with the slow vigilance that characterizes the cell's entire surveillance system, inspecting each point of contact with the double helix, as though reading its surface through touch alone. When a glycosylase encounters a damaged base, the recognition is subtle but decisive. Without disturbing the surrounding structure, the enzyme curls around the DNA and gently flips the affected base outward, lifting it away from its snug place within the helix. This movement is delicate, almost like turning a single page in a thick, ancient book. The base, now exposed, is removed from the sugar phosphate backbone, leaving behind a small, empty space, a site without a base. Known as an abasic site, the DNA remains intact, its backbone still whole, but one letter of its code has been gracefully plucked away. Once this small vacancy appears, another enzyme arrives, AP endonuclease. Its task is to recognize the missing base and create a tiny opening in the backbone, just enough for further repair steps to take place. This incision is precise, made with the care of a craftsman slicing along the edge of a delicate fabric. It does not unravel the helix nor disrupt the surrounding bases. It simply makes a small cut to allow access to the place where the missing base once rested. The opening is not chaotic but clean and intentional, an invitation to the next members of the cleanup crew. Now the cell brings forth DNA polymerase, an enzyme known for its role in replication but equally skilled at small repairs. Polymerase moves into the opening with gentle accuracy, reading the surrounding bases to determine which nucleotide should take the place of the one that was removed. Its work is methodical and patient. Using the intact strand of DNA as a guide, polymerase selects the correct base and inserts it into the empty space, restoring the proper pairing and re-establishing the gentle order of the helix. This is not a fast motion, but a precise one, each step based on the careful logic encoded in the structure of the DNA itself. Once the correct base has been restored, the sugar phosphate backbone must be sealed to complete the repair. This final act is performed by DNA ligase, an enzyme whose role resembles that of a quiet onlooker who steps in at the last moment to secure the final stitch in a piece of hand-sewn cloth. Ligase seals the small nick in the backbone. With a single, soft chemical bond, the DNA is made whole again, its code restored, its structure uninterrupted. Throughout this process, the atmosphere remains peaceful. All the movements are small and measured, carried out by proteins that drift through the nucleus with steady purpose. Nothing in base excision repair is rushed. The system is designed for calm correction, addressing the small changes that occur simply because the DNA is immersed in the living environment of the cell. The repair does not ask the cell to stop its activities. Instead, it unfolds quietly alongside them, just as the body maintains countless regulatory processes at once without drawing attention to any of them. The DNA is restored through a collaboration so subtle that the cell itself seems unaware of it, continuing its gentle rhythms of transcription, translation, and metabolic activity as the repair enzymes do their quiet work in the background. This process reflects a broader pattern found throughout biology: the tendency to maintain stability through small, constant acts of correction. Just as the body regulates temperature, glucose levels, and mineral concentrations through the cooperation of multiple systems, the DNA regulates its own integrity through the cooperation of multiple enzymes working in harmony. These actions are not reactive in the dramatic sense. They are anticipatory, addressing potential issues before they grow large enough to disrupt the cell's function. It is the biological equivalent of smoothing a wrinkle before it becomes a fold. As you continue to imagine the base excision repair system drifting through the nucleus, it becomes clear how remarkable this quiet cleanup crew truly is. It works constantly, addressing hundreds or even thousands of small irregularities each day, yet it does so without creating turbulence or strain. The DNA remains stable not because it avoids all damage, but because it is supported by a system that responds with unwavering calm. Each damaged base is removed, each small gap is repaired, each incision is sealed, all with movements so gentle that they resemble the soft care of someone sweeping a room with slow, deliberate strokes. In this way, the cell ensures that the memory of life remains clear and unbroken. The base excision repair system maintains the integrity of the DNA, not through force but through quiet attentiveness, preserving the soft harmony of the genetic code with the patient diligence of a caretaker tending a cherished garden. And in this ongoing, almost meditative process, the resilience of life reveals itself once more subtle, steady, and endlessly devoted to restoring balance whenever the smallest shift occurs. 7. Nucleotide excision repair, restoring larger regions. Within the soft interior of the cell, where the double helix drifts in gentle spirals, and the base excision repair system tends to the smallest imperfections with quiet diligence, there exists another layer of care designed for moments when the DNA experiences broader, more noticeable alterations. These disturbances are still not dramatic. They do not tear the helix apart or break it into fragments. Instead, they create small regions where the smooth, flowing surface of the DNA is slightly distorted patches where several bases have been nudged from their usual alignment by environmental influences or internal chemical shifts. To respond to these broader changes, the cell turns to a system known as nucleotide excision repair, a method that gently removes and replaces small stretches of damaged DNA so the double helix can return to its natural harmony. To imagine these disturbances, you might picture the DNA as a long, coiling ribbon held in a relaxed yet structured form. Its bases pair in serene uniformity, each one fitting into place like a bead in a long necklace. But when ultraviolet light touches the helix, or when certain chemicals in the environment interact with the bases, the surface of the ribbon can become slightly uneven. Thymine dimers formed when two adjacent thymine bases bond with each other under the influence of UV light are among the most common of these distortions. The effect is subtle but meaningful. The helix develops a small kink or bulge where the bases no longer sit properly against their partners. These are not catastrophic changes, but they do create areas where the DNA cannot be read smoothly by the cell's enzymes. The bases still rest within the helix, but their alignment has shifted just enough to require gentle correction. When such distortions arise, the cell's quiet surveillance network detects them with the same soft attentiveness it applies to smaller imperfections. Specialized proteins that scan for irregular shapes sense the altered curvature of the helix. These proteins do not need to decode the sequence to notice the problem. They simply feel the difference in the physical structure, much like fingers sensing an uneven thread in a woven fabric. Once a protein recognizes the distortion, it settles gently onto the DNA, marking the site for further inspection. Nothing about this process is rushed. The protein remains there, like a watchful guardian, standing beside a place that needs care, awaiting the arrival of the rest of the repair team. The first major member of that team is a multi-enzyme complex designed to verify the presence of a helix distortion. This complex moves with unhurried precision, sliding along the DNA until it reaches the marked site. Once it arrives, it examines the structure carefully, confirming that the region contains damage significant enough to require excision. Its presence stabilizes the DNA around the affected area, ensuring that the helix remains accessible and properly oriented for the next steps. This stabilization is gentle, almost like two hands cupping a delicate object to keep it steady while a repair is performed. Next, the cell brings forth helicase enzymes, proteins known for their ability to unwind DNA. These helicases do not separate the strands entirely. Instead, they create a small, open bubble around the damaged region. This unwinding allows the repair enzymes to access both strands of the DNA without disturbing the majority of the helix. The unwinding feels like a soft breath, gently lifting a small portion of the ribbon so the damaged area becomes more visible. The bases on each strand temporarily lose their partners in this region, but the surrounding structure remains peacefully intact, held in place by the stability of the remaining helix. Once the DNA has been opened slightly, endonucleases arrive to make precise incisions on either side of the damaged region. These enzymes cut the DNA not randomly, but at locations selected to remove the entire patch of distorted bases. The cuts occur several nucleotides away from the actual damage, creating a small excised section that includes the altered bases and a few of their neighbors. These incisions are delicate and controlled, resembling a gentle trimming of a frayed thread. or the removal of a slightly worn patch of fabric. The excised piece lifts away from the DNA, leaving a clean gap in the strand. This gap is not alarming, it is a carefully shaped space prepared for restoration. With the damaged region removed, DNA polymerase arrives to fill the empty space with new nucleotides. It reads the undamaged strand opposite the gap, using it as a template to ensure the new sequence matches the original. Polymerase moves slowly and steadily, adding one base at a time in the same quiet, orderly manner that allows it to replicate the entire genome when the cell divides. Each nucleotide it adds restores a small part of the helix's harmony, rebuilding the ribbon with the same gentle logic that guided its original formation. The repair is not rushed. Polymerase places each base with careful consideration, ensuring the restored region blends seamlessly into the surrounding DNA. Once the gap has been filled, the final step of the repair is performed by DNA ligase. This enzyme seals the last remaining nick in the sugar phosphate backbone, closing the repaired region with a single, soft chemical bond. Ligase's action is delicate but essential, completing the restoration by ensuring the helix once again forms a continuous, unbroken line. With this final touch, the DNA regains its smooth, even curvature, free from the distortion that had momentarily disrupted its symmetry. Throughout this entire process, the atmosphere within the cell remains peaceful. The DNA is never in danger. The distortions that prompted the repair are simply part of the natural way life interacts with its environment. Sunlight, mild chemical exposures, and the normal chemistry of living tissues create these small changes with gentle regularity, and the cell responds with equally gentle systems designed to maintain balance. The repair process unfolds quietly alongside the other rhythms of the cell: transcription, translation, and metabolism, without causing disruption or imbalance. It resembles the way the broader organism handles environmental changes, adjusting fluid levels, modifying metabolic rates, or altering gene expression in response to subtle shifts, all in an effort to maintain internal stability. What makes nucleotide excision repair so remarkable is not its complexity, though it involves many enzymes working in coordinated harmony. What stands out is its serenity. It does not tear or shred. It does not overwhelm the DNA with force. Instead, it removes only what is necessary, just a small patch of bases, and restores the area with precision and gentleness. The repair pathway exists as a testament to life's ability to maintain order through calm, patient correction, rather than dramatic intervention. As you picture this process, the soft unwinding of the helix, the careful removal of a tiny section, the slow rebuilding of the sequence, you may find the image soothing. It mirrors the way small disturbances in daily life can be addressed with quiet, steady actions, restored not through urgency but through thoughtful attention. The double helix, once more returned to its smooth, rhythmic form, continues to hold the memory of life, protected by a system that works with patience, subtlety, and unbroken devotion. 8. Mismatch repair after replication. Within the gentle stillness of the cell, where countless molecules perform their quiet tasks in unbroken harmony, there exists one process that unfolds with remarkable precision: the replication of DNA. This process allows the cell to divide, creating two daughter cells, each inheriting a complete copy of the genetic instructions that guide life's unfolding. Replication is astonishingly accurate, carried out by enzymes that read and pair nucleotides with careful consistency. Yet even in this calm choreography, small slips can occur, rare mismatches where a base pairs incorrectly, creating a tiny deviation from the intended sequence. These mismatches are not large distortions, nor do they break the helix. They are simply subtle errors, like a misplaced letter in a long, carefully written manuscript. And because the cell values harmony and precision, it employs a gentle but effective system mismatch repair to identify and correct these errors shortly after replication has taken place. To imagine mismatch repair, it helps to think of the replication process itself. As the helix unwinds, Each strand serves as a template for forming a new complementary strand. Polymerase, the enzyme responsible for adding bases, moves along each template with a steady rhythm, selecting the correct nucleotide based on the rules of pairing. Adenine with thymine, cytosine with guanine. It performs this task with astonishing accuracy, placing each base as though it were placing a bead onto a string, ensuring that the growing strand mirrors the original with remarkable fidelity. Yet even the most precise system encounters occasional slips. A base may be incorporated that does not correctly match the template. Perhaps an adenine is paired with a cytosine, or a guanine with a thymine. These mismatches create slight irregularities in the helix, subtle enough that polymerase may not always detect them immediately. When replication is complete, the DNA rests in its newly formed state, with the freshly synthesized strands paired alongside their original templates. It is in this moment, quiet, transitional, and full of potential, that mismatch repair begins its gentle sweep. The cell sends out specialized proteins that drift along the newly replicated DNA, searching for the soft distortions that mismatched pairs create. These proteins sense not the sequence itself, but the shape, feeling for slight deviations in the helix's smooth curvature. A mismatched base pair bulges ever so slightly, or creates a faint ripple, and the surveillance proteins detect this change the way fingertips might detect the smallest snag in a piece of silk. Once a mismatch is recognized, the repair process begins with uncanny precision. The first challenge is determining which base is incorrect. Because DNA replication produces two strands, one old and one new, the cell must identify the newly synthesized strand where the error occurred. This distinction is possible because newly made DNA carries subtle marks, small chemical cues that fade with time but remain present long enough to guide the repair system. In bacteria, for example, the older strand is methylated. In more complex organisms, the distinction arises from the way the replication machinery leaves behind temporary signals along the new strand. These gentle markers allow the mismatch repair system to identify the strand that requires correction, ensuring the original template is preserved and used as the reference for accurate repair. Once the system identifies the incorrect strand, an enzyme binds to the region surrounding the mismatch. It stabilizes the helix, holding it gently while additional proteins are recruited. These repair proteins work together, forming a soft assembly around the error, their presence almost like a quiet circle of caretakers gathering around a single point that needs tending. The DNA loops slightly to allow the proteins to interact with the mismatch, creating a small, localized bend that does not disturb the overall serenity of the helix. Then, the repair enzymes begin their work. An endonuclease makes a precise incision in the newly synthesized strand, not directly at the mismatch, but some distance away. This cut creates an entry point for an exonuclease, an enzyme that begins removing nucleotides from the strand. It moves toward the mismatch in a slow, steady pace, excising the section of DNA that contains the incorrect base. This removal is not abrupt. It is methodical, resembling the gentle, unraveled pulling of a thread from a fabric, allowing the cell to discard the incorrect portion while preserving the integrity of the rest of the molecule. Once the mismatched region is removed, the DNA is left with a small gap where nucleotides must be replaced. DNA polymerase steps in again, performing its familiar role of rebuilding the strand using the original template as a guide. Because the opposite strand carries the correct sequence, the polymerase can restore the missing bases with calm accuracy, inserting the appropriate nucleotides one by one until the strand is complete. Each addition restores a bit more harmony to the helix, re-establishing the correct pairing that ensures the genetic instructions remain intact. Finally, DNA ligase arrives to seal the nick in the sugar phosphate backbone, completing the repair with a final, gentle touch. Ligase's role is always quiet but essential, closing the gap so the DNA once again forms a smooth, continuous structure. With the mismatch corrected and the backbone sealed, the repair system releases the DNA, The entire mismatch repair process unfolds swiftly and silently, usually within moments of replication. It does not interrupt the cell's other activities nor create strain or urgency. Instead, it reflects the cell's desire to maintain fidelity in the genetic code. Without mismatch repair, the rate of replication errors would increase dramatically. But with this quiet system in place, the errors remain rare, corrected long before they can disturb the flow of life. This gentle correction process also reflects one of the central principles of biological organization: the interplay between variation and stability. While natural selection shapes organisms through heritable differences, the cell simultaneously works to preserve the integrity of its genetic material, ensuring that only meaningful and beneficial variations persist through evolution. Mismatch repair plays a role in this balance, preventing accidental errors from overwhelming the orderly structure of the genome. As the newly corrected DNA settles into the stillness of the nucleus, it becomes clear how deeply the cell values harmony. Mismatch repair, though intricate in its details, embodies a simple truth. Life depends on both change and consistency, on the gentle interplay between replication's creative act and the quiet systems that preserve accuracy. The double helix continues its slow drift within the nucleus, restored to its even form by a repair process that works with the soft persistence of a caretaker brushing away the smallest of disruptions. Here, in the quiet after replication, life continues forward, guided by the steady hands of molecular guardians who ensure each new strand carries the clarity and balance needed for the cell to thrive. 9. Double-strand breaks. The cell's calm response to serious disruptions. In the quiet landscape of the cell, where the double helix flows in gentle spirals and the smaller imperfections of life are handled with steady care, there occasionally arises a different kind of disturbance, one that affects not just a single base or a small patch of nucleotides, but both strands of the DNA at once. These are known as double-strand breaks, and although they may sound dramatic when described in words, within the serene environment of the nucleus, they emerge not as sudden shattering events, but as soft interruptions in the continuity of the molecule. The helix does not collapse or twist apart wildly. It simply encounters a moment where both strands are cut or disrupted, leaving a small gap that must be addressed with greater attention than the more common, gentler forms of DNA wear. Yet even in these moments, the cell responds with calm deliberation, using carefully orchestrated pathways that restore integrity with the same quiet precision seen throughout cellular life. To picture a double-strand break, you might imagine the DNA as a long and delicate ribbon suspended in still water. Most of the time it remains intact, curving in smooth loops and maintaining its stable shape. But occasionally, due to naturally occurring processes or interactions with certain environmental factors, a small section of the ribbon may be gently severed, leaving two free ends that drift ever so slightly apart. These breaks can arise from a variety of soft influences, from the byproducts of metabolic activity, to low-level radiation, to rare slips during replication or recombination. None of these causes occur with violence inside the cell. Rather, they represent the natural vulnerability of a molecule that must remain dynamic, open to being copied, read, and adjusted. Double-strand breaks simply reflect the reality that even the strongest biological structures sometimes require more significant forms of repair. When such a break occurs, the first response of the cell is one of recognition. Specialized proteins sense the presence of free DNA ends, which differ from the continuous, unbroken shape of an intact helix. These proteins move toward the break with the same patient curiosity that characterizes the rest of the cell's surveillance network. They bind gently to the exposed ends, not sealing them but stabilizing them, preventing further fraying or unwanted interactions. This initial binding is often carried out by proteins such as the mRNA complex in eukaryotic cells, a trio of molecules that forms a calm, steady presence around the break. The complex does not rush to act. Rather, it creates a protected space, ensuring the broken ends do not drift apart or become chemically reactive. In this sense, the mRN complex acts much like a caretaker placing soft hands around a cracked but cherished object, holding it in a safe position so the repair can proceed without further harm. Once the break has been recognized and stabilized, the cell must choose which repair pathway to use. There are two primary methods: homologous recombination and non-homologous end joining. Each pathway carries its own subtle logic, its own sense of suitability depending on the cell's condition and the phase of the cell cycle. Yet neither pathway is chosen with urgency. The choice arises naturally from the state of the DNA and the presence or absence of certain molecular markers. The cell, in its quiet intelligence, simply selects the approach that fits the moment, much like the body as a whole adjusts its responses to maintain balance across its organ systems. Homologous recombination is the more precise of the two pathways, often used when a sister chromatid is available as a template, typically after the DNA has been replicated but before the cell divides. This pathway begins with a gentle resectioning of the broken ends. Certain enzymes trim back the ends of the DNA strands, creating soft, single-stranded overhangs that begin to drift slightly outward. These overhangs are then coated with a protein called Rad51, which wraps around them like a calm protective sheath, guiding the next steps with quiet precision. The coated strands then search for a matching sequence on the intact sister chromatid, a process that unfolds with patient exploration. When a matching region is found, the broken strand gently invades the intact helix, pairing with its complementary sequence. This pairing allows the cell to use the intact chromatid as a guide to rebuild the damaged region, ensuring that the repair is faithful to the original sequence. Enzymes move along the paired strands, extending the broken ends and using the template to restore missing nucleotides. Eventually, the newly synthesized segments are rejoined, and the DNA untangles itself from the sister chromatid, returning to its smooth, continuous form. Homologous recombination represents one of the most serene and elegant repair mechanisms in biology, using the quiet redundancy inherent in DNA replication to guide restoration. Non-homologous end joining, in contrast, is a simpler and more direct pathway, used when no template is available. In this method, the broken ends of the DNA are brought together and gently aligned. Specialized proteins hold the ends in place, trimming any damaged nucleotides and preparing the strands for rejoining. Once the ends are clean, a ligase enzyme seals them together, completing the repair. This method is not as precise as homologous recombination, and small changes in the sequence may result. Yet even this less exact pathway unfolds with calm coordination, ensuring that the DNA remains intact and functional. It reflects the cell's preference for continuity, choosing to preserve the structure of the helix even if a few bases must be altered to achieve closure. Throughout these repair processes, the cell maintains a sense of quiet order. The pathways do not conflict or compete. They unfold according to the needs of the moment, guided by molecular cues that subtly influence each step. The enzymes involved do not rush or strain. Their movements are smooth and deliberate. Each action, whether trimming, aligning, or synthesizing, contributes to the restoration of harmony within the nucleus. Even in the face of disruptions that affect both strands of the helix, the cell responds not with urgency but with steady competence, reflecting a deep resilience that permeates every level of biological organization. Double-strand breaks, despite their potential seriousness, are not treated as emergencies within the cell. They are simply another type of irregularity, a different kind of shape that the surveillance network must recognize and address. The repair pathways are available not because the cell expects disorder, but because it accepts the natural flow of change that occurs within living systems. Just as the body maintains balance through countless small adjustments, the DNA maintains its integrity through processes that value patience, precision, and quiet cooperation. As the broken strands are restored, whether through recombination or end joining, the DNA gradually regains its even curvature. The helix settles once more into its softly spiraling form, drifting in the serene environment of the nucleus as though nothing had happened. The cell moves forward, continuing its gentle cycles of reading, copying, and maintaining life's instructions. And the double-strand break, once a small pause in the flow of the genome, becomes just another example of how life responds to disruption with calm, deliberate care. In this way, the cell's response to double-strand breaks reflects a profound truth. Even the most significant interruptions in life structure can be met with quiet resilience. The DNA, supported by its molecular guardians, returns to harmony not through force or urgency, but through steady, thoughtful repair, a reflection of the deep calm that underlies all living systems. 10. Homologous recombination. Borrowing a safe template. In the soft interior of the cell, where DNA drifts like a quiet ribbon of memory and countless repair systems move with unhurried precision, there is a repair process that stands out for its extraordinary accuracy and calm coordination. This process, known as homologous recombination, is the cell's most faithful way of repairing double-strand breaks, those moments when both strands of the DNA molecule experience an interruption in continuity. Although such breaks may seem serious when described in words, within the serene environment of the nucleus, they are treated as gentle pauses in the natural flow of the helix, disturbances that call for patient healing rather than alarm. Homologous recombination steps into this role with the same deliberate grace that characterizes all of the cell's repair mechanisms, offering a method of restoration that relies on the presence of a safe, undisturbed template. To understand homologous recombination, it helps to imagine the cell at a moment after DNA replication has occurred but before cell division begins. During this window, the cell contains two nearly identical copies of its DNA, each one consisting of a newly formed strand paired with an older strand. These copies, known as sister chromatids, rest close together in the nucleus, coiled gently around their associated proteins and drifting in the subdued rhythms of chromatin. Their proximity is not coincidental. It reflects the cell's understanding that having a duplicate copy of its genetic information provides a unique opportunity for precise repair. When a double-strand break occurs during this stage, the cell's repair systems recognize the availability of an intact template and choose homologous recombination as the most accurate pathway for restoration. The process begins with recognition, the moment when the cell's surveillance proteins detect the broken ends of the DNA. These proteins, often forming part of a complex network such as the mRNA complex, bind gently to the free ends, stabilizing them so that no further fraying or chemical interference can occur. The presence of these proteins is not forceful. It is more like a calm embrace that holds the broken molecule steady. The cell does not seek to rush this moment. Instead, it takes time to prepare the strands for the next steps, ensuring that the environment around the break is peaceful and well organized. Once the ends have been secured, the repair enzymes begin a process known as end resection. In this stage, small sections of the DNA surrounding the break are trimmed away, exposing single-stranded regions at the broken ends. These single-stranded segments extend like delicate threads, drifting slightly away from the main structure of the helix. They are not left unprotected for long. Specialized proteins quickly bind to them, coating the single-stranded DNA to prevent it from curling back on itself or attaching to unwanted regions. One of the most important of these proteins is Rad51, which assembles along the single-stranded regions like a soft sheath, guiding them through the next steps with patient direction. With Rad51 bound, the single-stranded DNA begins its quiet search for a matching sequence. This search unfolds within the gentle space of the nucleus, where the sister chromatids float in proximity. The coded strands move with slow, careful motions, scanning the undisturbed DNA for a region that mirrors their own sequence. When they find the correct location, and they almost always do, the single-stranded DNA threads slip between the paired strands of the intact chromatid. This moment, known as strand invasion, is the heart of homologous recombination, It is a process so elegant and subtle that it is often compared to the joining of two matching ribbons, where one ribbon gently weaves into the folds of another, guided by their shared pattern. Once the invaded strand pairs with the intact template, the repair machinery begins the process of rebuilding the missing information. DNA polymerase, the same enzyme that replicates the genome, moves along the invaded region, synthesizing new DNA to replace what was lost in the break. The polymerase uses the intact strand as a guide, ensuring that each nucleotide it adds matches the original sequence. This restoration unfolds with soft precision, like a painter slowly restoring a section of an old mural by matching colors and shapes with quiet concentration. The cell does not rush polymerase. Instead, it lets the enzyme place each base with confidence, knowing that accuracy is the foundation of the helix's harmony. As the new DNA is synthesized, the strands begin to unwind from the sister chromatid, gradually returning to their original location. But they do not do so abruptly or in haste. The molecules slide apart with the same gentle logic that guided their pairing, returning to their respective positions while bringing with them the newly restored sequences. Depending on the specific pathway of homologous recombination, the strands may form intermediate structures, such as holiday junctions, that look like small, elegant crossovers of the DNA. These junctions are resolved slowly and precisely by specialized enzymes, which separate the strands without disrupting the surrounding chromatin. The result is a repaired molecule that matches the original sequence with remarkable accuracy, its continuity restored through an act of quiet molecular cooperation. The importance of homologous recombination becomes even clearer when one considers its role in maintaining genetic stability. Unlike other repair pathways that sacrifice a small amount of accuracy for speed or simplicity, homologous recombination prioritizes fidelity above all else. It ensures that the repaired region mirrors the original sequence, preserving the integrity of the genetic code. This fidelity is essential not only for the health of the cell but for the organism as a whole, as it allows genetic information to remain consistent across countless cycles of replication and division. This repair mechanism also reflects the broader principle of redundancy in biology. Just as organisms maintain multiple organ systems that support and balance each other, cells maintain multiple copies of certain molecules or pathways to ensure reliability. The presence of sister chromatids provides an extra layer of security, giving the cell a backup copy that can guide precise repair. This kind of redundancy is not wasteful. It is a hallmark of biological resilience, allowing life to persist even in the face of fluctuations or unexpected changes. The gentle interplay between the copies in homologous recombination mirrors the harmony found throughout biological systems, where balance emerges from cooperation. The serene nature of homologous recombination also highlights how deeply the cell values continuity, rather than discarding damaged regions or patching them in ways that might introduce small variations. The cell chooses a pathway that restores the sequence with near-perfect accuracy. It takes the time required to ensure that the restored DNA remains true to the original blueprint. This commitment to restoration reflects the quiet patience that defines cellular life. A patience shaped by billions of years of evolution and refinement. As the repaired double helix once again settles into its peaceful curvature, drifting in the gentle currents of the nucleus, the cell resumes its quiet routines. Transcription resumes, replication continues when needed, and the DNA once again serves as the calm repository of life's instructions. The momentary disturbance caused by the double-strand break becomes part of the cell's history, a brief deviation corrected through the unwavering devotion of homologous recombination. In this way, homologous recombination stands as one of the cell's most elegant forms of care. It is a pathway rooted in calm precision, guided by the presence of a safe template and a deep respect for the integrity of the genetic code. Through it, the cell restores its most precious molecule with the gentleness of a craftsman repairing a cherished heirloom, returning the double helix to its natural harmony with quiet, steady hands. 11. Non-homologous end joining. Quiet efficiency. Within the gentle interior of the cell, where countless molecular rhythms unfold in calm coordination, not every moment of repair requires the elaborate precision of homologous recombination. Sometimes the cell must respond to a double-strand break when no sister chromatid is available, or when the break occurs during a phase of the cell cycle where the genome has not yet been duplicated. In these moments, the cell turns to a different form of care, a pathway known as non-homologous end joining, or NHEJ. While this method does not rely on a detailed template and may not restore the DNA with perfect accuracy, it embodies a beautiful kind of quiet efficiency, sealing broken strands with the steady, unhurried motion of a caretaker who knows that even imperfect repairs can preserve the delicate continuity of life. To imagine how non-homologous end joining unfolds, it helps to return to the soft image of DNA drifting like a ribbon through the calm fluid of the nucleus. A double-strand break interrupts this ribbon, creating two free ends that float gently apart. These ends are stable for only a brief moment before the cell's sensing proteins detect their presence. In the stillness surrounding the break, one of the first proteins to arrive is known as COO, a pair of molecules shaped so they can slide over the exposed ends like protective caps. COO binds to the DNA without haste, settling over each end with quiet certainty. Its presence prevents the ends from fraying or attaching to the wrong partners. It does not attempt to close the break by itself. Instead, it simply holds the ends in place, creating a zone of stability around the site. The arrival of COO sets the stage for the rest of the repair process. This protein acts as a calm signal, inviting other repair factors to gather. One of the next to arrive is DNA PKCs, a large protein kinase that attaches to COO and forms a protective complex around the broken ends. Together, these components bring the two ends into closer proximity, aligning them so that they face one another across the small gap. This alignment is not sharp or forceful. Rather, it resembles the gentle adjustment of two pieces of fabric being positioned for stitching. The cell does not insist on perfect alignment at this stage. It simply seeks to bring the ends close enough that they can eventually be joined. With the broken ends stabilized and aligned, the cell must prepare them for rejoining. Breaks do not always occur in neat, even cuts. Sometimes the DNA ends have overhangs. Sometimes they carry small chemical modifications. Sometimes a nucleotide or two has been lost from one or both strands. To smooth these irregularities, the cell brings in enzymes that can gently trim or fill in the ends. Artemis, an important nuclease in this pathway, acts like a soft-edged tool that can remove misplaced or damaged nucleotides. Other enzymes, such as specialized polymerases, fill in missing sections with new nucleotides, guided by subtle cues that keep the additions small and controlled. These actions resemble the quiet preparation of a surface before a final adhesive is applied, cleaning, smoothing, and shaping the edges so they can fit together as cleanly as possible. Throughout this preparation, the entire process remains steady and unhurried. The cell knows that it cannot restore the DNA with the level of precision offered by homologous recombination in these moments, yet it does not treat the situation with alarm. Instead, it adheres to a simple, effective pattern: stabilize, align, prepare, and seal. This rhythm echoes the way many forms of biological regulation unfold, from the subtle balancing of ions that maintains the body's internal environment, to the gentle shifts in hormone levels that guide growth, development, and daily physiological rhythms. Order arises not from frantic correction, but from calm, continuous adjustment. Once the ends of the DNA are smooth and properly oriented, the final and essential step begins. DNA ligase 4, accompanied by its cofactor XRCC4, moves toward the break. This enzyme is the one that completes the process, sealing the two strands together by creating the chemical bonds needed to restore the continuity of the backbone. Ligase works with a kind of serene confidence, touching the ends lightly, making sure they sit comfortably against one another, and then forming the covalent bonds that hold them in place. Its movement is almost ritualistic in its simplicity. There is no need for complex sequences or intricate recognition patterns. Its task is simply to bring two ends together and quietly complete the repair. The moment the ligase forms the final bond, the DNA returns to a continuous form. It may bear a few subtle changes in its sequence, perhaps a missing base, or a slight shift in the order of nucleotides. Yet these small variations often have no effect on the cell's overall health. The helix resumes its gentle curvature, floating once more in the serene environment of the nucleus, ready to guide the cell through its normal rhythms of transcription and replication. The break becomes a memory, resolved through the quiet competence of the NHEJ pathway. What makes non-homologous end joining so remarkable is the sense of sufficiency that defines it. The cell does not require perfection in every moment. It understands that most of its DNA can withstand small changes without disrupting the flow of life, and so it uses NHEJ as a practical, calm solution, a way to maintain continuity, even when the exact sequence cannot be preserved. In this sense, the pathway mirrors broader biological truths. Organisms constantly adapt to small variations, and the genome itself accumulates changes through evolution, allowing life to diversify across generations. The work of NHEJ does not disrupt this process. It participates in the natural interplay between stability and change, allowing the DNA to move forward with its integrity mostly intact and its essential structure preserved. Non-homologous end joining also reflects the ways in which biological systems embrace flexibility. The body does not insist on perfect symmetry or ideal conditions to function. It compensates, adapts, and finds workable solutions through layers of regulation that unfold with quiet patience. The immune system adjusts its responses based on daily encounters. The digestive system adapts to different foods. The nervous system recalibrates itself throughout life. Similarly, NHEJ represents an adaptable response to a potentially serious form of DNA damage. It may not produce flawless results, but it restores continuity, allowing the cell to remain whole. As you picture the DNA after a repaired break, sealed through the calm work of ligase and held steady by the proteins that previously guided the alignment, you may sense the gentle resilience inherent in the genome. Whether through precise recombination or through efficient end joining, the DNA is supported by systems that value continuity, harmony, even in the face of interruptions that affect both strands of the helix. The cell maintains its composure and responds with methods that preserve the underlying stability of the organism. In this quiet capacity to mend serious disruptions without haste or anxiety, non-homologous end joining reveals itself as one of the many soft strengths of cellular life, a pathway rooted in gentle efficiency, guided by the simple truth that restoring connection is often more important than restoring perfection. 12. Cell cycle checkpoints, gentle pauses for safety. Within the calm interior of the cell, where DNA drifts in its soft spiraling form and repair systems move with patient precision, there exists another protective layer, one that does not directly mend the DNA, but instead guides the entire cell through its moments of growth and preparation. These are the cell cycle checkpoints, quiet pauses built into the rhythm of cellular life. They act like gentle resting places on a long journey, moments where the cell slows its pace, evaluates its internal conditions, and ensures that everything remains in harmony before moving forward. Rather than rushing from one phase to the next, the cell follows a pattern of thoughtful progression, each step guided by signals that confirm whether the DNA is intact, properly prepared, and ready to be carried into the future. To imagine these checkpoints, you might begin by picturing the cell cycle itself, not as a rigid sequence, but as a soft circle of stages through which the cell moves with steady rhythm. The first stage, known as G1, is a peaceful period of growth and gentle metabolic activity. The DNA rests in its unreplicated state, while the cell gathers nutrients, evaluates its conditions, and prepares the environment for replication. The next stage, S phase, is where DNA replication occurs, with the entire genome copied in a long, quiet sweep. Then comes G2, another reflective stage where the newly duplicated DNA is reviewed and prepared for the complex process of division. Finally, the cell enters mitosis, where the chromosomes are separated and the organism's most fundamental act of continuity, cell division, takes place. Throughout this cycle, checkpoints appear like soft gateways, quietly asking the cell simple but essential questions. Is the DNA ready? Has it been properly copied? Are any repairs needed before proceeding? These checkpoints do not display urgency or alarm. Instead, they create small pauses, soft moments of stillness where the cell can make adjustments before moving forward. They are the guardians of timing, the keepers of balance, ensuring that each step unfolds in harmony with the whole. The first major checkpoint appears at the end of G1, before the DNA begins to replicate. At this moment, the cell gently evaluates its surroundings. It senses the availability of nutrients, the integrity of its existing DNA, and the overall environment within the organism. If conditions are calm and healthy, the cell receives a soft molecular signal, allowing it to move into S phase. But if the DNA is damaged, perhaps containing lesions or mismatches from natural sources of everyday wear, the checkpoint offers a quiet pause. It does not force the cell forward. Instead, it allows the cell to remain in G1, while repair systems address any irregularities. The checkpoint is not a block but a resting place, giving the cell the time it needs to restore the integrity of its genetic material. Once replication begins in S phase, the DNA undergoes a remarkable transformation. The double helix unwinds, polymerases move along its strands, and two complete copies of the genome emerge in slow, orderly fashion. Throughout this process, another layer of watchfulness persists, ensuring that the replication machinery proceeds in a stable, unhurried rhythm. If the replication fork encounters a lesion or a difficult region, the cell senses the slowdown and engages mechanisms that protect both the template and the newly forming strands. Specialized proteins stabilize the replication machinery, allowing time for repair enzymes to correct any mismatches or distortions that arise. This form of checkpoint activity reflects the cell's deep patience. It slows replication rather than risking the creation of errors, preserving the integrity of both strands. As the cell completes replication, it enters G2, a stage filled with quiet preparation. The DNA now exists as sister chromatids, genetically identical copies that rest side by side, coiled with serene symmetry. The cell gently checks for any remaining structural irregularities, ensuring that both chromatids are whole and properly paired. If a double-strand break occurred during replication, this is the moment when homologous recombination can step in, using one chromatid as a template to restore the other. The G2 checkpoint remains open only when the DNA is intact, its structure smooth and relaxed. But if the cell detects damage, a small distortion, a gap, or an incomplete region, it holds the cycle in a soft pause, allowing repair pathways the time and space they need to restore harmony. One of the most profound checkpoints appears just before the cell enters mitosis. In this moment, the cell must be certain that the DNA is properly duplicated and free of errors. For mitosis requires precision. The chromosomes condense into tighter structures, preparing for the elegant choreography of separation. The checkpoint at this stage acts like a final gentle guardian, ensuring that no damaged or incomplete DNA moves into the division process. If needed, the cell waits, repairing any irregularities before continuing. Once mitosis begins, another checkpoint emerges at the metaphase stage, where chromosomes line up at the center of the cell. This checkpoint ensures that each chromosome is properly attached to the spindle apparatus that will guide its movement. The cell does not move forward unless every chromosome rests in place, aligned with the soft symmetry that mitosis requires. If one chromosome drifts slightly or fails to attach, the checkpoint responds with stillness, holding the cell in a calm, suspended moment while adjustments are made. Only when perfect alignment is reached does the cell proceed, dividing its chromatids into two future daughter cells. Throughout all of these stages, the checkpoints do not operate as strict barriers, but as thoughtful pauses. They offer the cell time to repair, time to stabilize, time to ensure that its most precious molecule remains whole. They reflect a deep respect for the continuity of life, ensuring that each generation of cells inherits DNA that is as complete and accurate as possible. Without these pauses, cells would rush through the cycle, replicating damaged DNA or dividing with structural irregularities. But with checkpoints in place, the cell maintains a sense of rhythm that aligns with the quiet harmony of all its internal processes. The checkpoints also serve as a reminder of how living systems function, not through constant speed or perfection, but through balance, reflection, and careful response to change. The body maintains stability through homeostasis, adjusting small variations in temperature, hydration, and metabolic activity with slow, steady control. Similarly, the cell maintains genomic stability by giving itself moments of stillness, spaces where repair can occur without disruption. In these pauses, the cell embodies patience. It does not push forward until conditions are right, and when conditions are not right, it simply waits. This waiting is not passive, it is filled with soft activity, as enzymes glide along the DNA, repairing mismatches, restoring breaks. The checkpoints create the time needed for these quiet efforts, allowing the cell to remain resilient in the face of natural fluctuations. As you imagine the cell moving through its cycle, guided by these gentle stopping points, the entire process begins to feel like a long, slow breath. Inhale, pause, exhale, pause, each phase unfolding with a sense of rhythm and calm. The DNA flows through moments of replication, repair, and division, never hurried, always supported by systems designed to protect its harmony. Even in the complexity of cell division, the overall impression is one of softness, reflection, and unbroken continuity. In this way, the cell cycle checkpoints stand as quiet guardians, ensuring that each step of cellular life unfolds in balance, clarity, and gentle wisdom. They create the space for healing, the time for care, and the opportunity for each new generation of cells to begin with DNA that has been tended with patience and devotion. 13. Polymerases, ligases, and other soft-spoken helpers. Inside the gentle stillness of the nucleus, where DNA rests like a spiraling thread of memory, and the cell's many repair systems glide in soft, deliberate motions, the act of maintaining the genome depends on a diverse array of enzymes. These molecules, each shaped by evolution to fulfill a specific role, move with a kind of understated grace, performing some of the most essential tasks required for life. They do not rush, nor do they rely on force. Instead, they act with quiet assurance, allowing the delicate structure of the double helix to be copied, restored, and preserved. Among these helpers, DNA polymerases and DNA ligases stand as two of the most familiar, yet they are joined by many other proteins, each contributing to the soft network of care that keeps the genome whole. To imagine these enzymes at work, you might picture the DNA as a long and luminous ribbon drifting through a calm interior world. Along this ribbon, various molecules pass with rhythmic steadiness, like caretakers tending to an ancient manuscript. They are mindful, attentive, and gentle, recognizing the importance of every nucleotide, every bond, and every structural curve. The environment around them is quiet, the motions smooth, and the outcomes precise. DNA polymerases are among the most dedicated of these helpers. They carry the responsibility of copying the genetic material during replication, a task that requires extraordinary fidelity, Yet, despite their accuracy, polymerases do not feel hurried. When they move along the DNA, they glide with a patient and almost meditative rhythm, reading the sequence of bases and selecting their complementary partners with soft determination. Each time a polymerase adds a nucleotide to a growing strand, it does so with a moment of verification, ensuring that the fit is correct. The base pairs form with a delicate snap of hydrogen bonds, quiet, almost inaudible, yet essential to maintaining the proper structure of the helix. These enzymes also play a central role in repair. When small gaps appear or damaged regions are excised, polymerases are called to the site. Their task is to fill these spaces with the correct nucleotides, guided by the undamaged strand that lies opposite. They perform this act of restoration as though they are gently reinforcing the pattern of a woven fabric, ensuring that each new thread matches the one that came before. Whether they are filling a single missing base during base excision repair or rebuilding larger regions after mismatch repair or nucleotide excision repair, polymerases approach their work with the same calm, deliberate process. They bring order to the places where disruption has occurred, but polymerases alone cannot complete the repair. Once a region has been rebuilt, there remains a small, narrow gap in the backbone, a nick where the sugar phosphate chain is not yet fully connected. This is where DNA ligase enters, a quiet closer of seams, a soft finisher of lines. Ligases operate like careful artisans, bridging the final space between two nucleotides with a single covalent bond. This act is almost ceremonial in its simplicity, the final touch that transforms an open, incomplete structure into one that is whole again. Ligases do not need to move quickly. Their job is one of precision and continuity. They ensure that every repaired strand becomes once more an unbroken thread, ready to flow seamlessly through the nucleus. Around these two central helpers exist many other enzymes, each with their own calm role in maintaining the DNA. Exonucleases, for example, remove nucleotides from the ends of DNA strands. They do not tear away large segments but nibble gently, trimming only what is necessary to create clean edges for repair. Their work resembles the soft smoothing of a frayed ribbon, preparing it to be joined once again. Endonucleases, in turn, are like quiet sculptors. They cut the DNA at precise internal locations, not to harm the helix, but to help remove damaged regions or to create the clean boundaries needed for repair pathways to function. Their incisions are controlled and measured, guided by the recognition of specific structural cues. The cuts they make are deliberate, small, and essential, allowing the next steps in repair to unfold smoothly. Other proteins act as guides rather than cutters. Helicases, for instance, unwind sections of the DNA so that repair enzymes can access the bases underneath. Their motion is slow and purposeful, separating the strands without tearing or straining them. They gently ease the helix open, like someone turning the pages of a delicate book. The double helix responds to their touch with subtle elasticity, unwinding, and then returning to its natural form once the repair is complete. Single-strand binding proteins also play a quiet but crucial role. When the helix is unwound, the single strands can feel exposed and unstable, prone to forming loops or collapsing back on themselves. These proteins move in like soft stabilizers, attaching to the single strands and holding them gently apart. They protect the exposed bases, preserving their structure until the polymerases can rebuild the missing or damaged regions. Their presence is almost comforting, a molecular embrace that keeps the strands safe during vulnerable moments. Repair scaffolding proteins act like coordinators, bringing together multiple enzymes so that they can work in harmony. They do not perform the repair themselves, but facilitate the interactions that make repair efficient. Their presence is reminiscent of quiet organizers in a peaceful workshop, those who arrange tools, align materials, and ensure that each artisan has what they need to work without interruption. Even the histone proteins that package the DNA participate in this calm orchestration. When repair must occur, certain histones loosen their hold, allowing the chromatin to open slightly so that enzymes can access the DNA. After the repair is complete, the histones help rewrap and reorganize the helix. Their actions are subtle but essential, forming the structural foundation that supports all other activities within the nucleus. These enzymes, though numerous and diverse, share a common thread. They operate with quiet cooperation. None of them dominates the process. Instead, they interact through soft signals, protein-protein contacts, and local chemical cues. Their rhythms align without conflict, creating an environment where the DNA is tended with care, patience, and unfailing devotion. Each enzyme fulfills a specific role, but together they form a collective presence, an ensemble of soft-spoken helpers whose shared purpose is to guard the integrity of the genetic material. Their work also reflects the interconnected nature of biological systems. Just as the body relies on cooperation among its organ systems to maintain homeostasis, the DNA relies on cooperation among its repair enzymes to maintain stability. The digestive system, the respiratory system, the circulatory system, each contributes to the balance of the organism. Similarly, polymerases, ligases, helicases, nucleases, and structural proteins contribute to the balance of the genome. Their functions overlap, complement, and support each other, forming an internal harmony that sustains life at the molecular level. As you imagine these enzymes drifting softly along the DNA, repairing, restoring, unwinding, sealing, and stabilizing, the overall effect becomes deeply soothing. Within the quiet space of the nucleus, where every movement is measured and every interaction purposeful, the DNA is tended by hands that never tire, never rush, and never lose their attentiveness. These enzymes embody the patience inherent in living systems, the patience that allows cells to maintain their integrity through countless cycles of replication and renewal. And so the double helix continues its slow, peaceful drift within the nucleus, supported by an entire ensemble of helpers whose quiet voices shape the future of the cell. Through their gentle efforts, the DNA remains whole, resilient, and ready to guide the next moment of life with its soft and unwavering clarity. 14. Chromatin remodeling, opening the shelves of the genetic library, deep within the nucleus, where the DNA rests in its soft spiraling form and repair systems glide with gentle purpose. The genetic material is never simply floating freely. Instead, the long strands of DNA are wrapped around proteins called histones, forming structures known collectively as chromatin. This winding creates organization, protection, and order, much like shelves in a quiet library holding ancient scrolls. Yet, whenever the cell needs to read a particular section of DNA or repair a region that has been slightly worn or disrupted, the chromatin must shift, loosen, and reorganize. This process, known as chromatin remodeling, is one of the calmest and most elegant mechanisms in the cell, unfolding with the grace of a caretaker gently opening a book that has rested unopened for many years. To imagine chromatin, think of the DNA as an incredibly long ribbon wrapped again and again around small, rounded spools. Each spool is a cluster of histone proteins, and together, the DNA and histones form structures called nucleosomes. These nucleosomes repeat along the length of the DNA like beads on a quiet string, creating a gentle pattern that keeps the genetic material compact and safe. This organization prevents tangling, protects regions from unnecessary chemical interactions, and ensures that the DNA remains stable within the soft environment of the nucleus. Yet this compactness also means that the DNA is not always immediately accessible. Just as a scroll must be unrolled before its words can be read, a region of DNA must be unwound from its histones before repair enzymes or transcription machinery can reach it. Chromatin remodeling provides the answer to this need. It does not unfold the entire genome at once, only the specific regions that require attention. The rest of the chromatin remains in its peaceful, organized form. when the cell senses that a particular region of DNA must be accessed, whether for repair, replication, or transcription. Chromatin remodelers move in with calm and deliberate precision. Chromatin remodelers are proteins or complexes of proteins designed to adjust the way DNA interacts with histones. They use small amounts of energy, not to push or tear, but to gently nudge the nucleosomes along the DNA, or to loosen the contacts between the DNA and the histone cores. Their actions are subtle, resembling the soft adjustments made by a librarian sliding a book forward on a shelf, lifting it only enough to reveal the pages within. These remodelers do not disrupt the overall architecture of the chromatin. They simply create small openings, spaces where other proteins can slip inside to perform their tasks. One of the most common forms of chromatin remodeling involves sliding nucleosomes along the DNA. In this process, the remodeler binds to a nucleosome and slowly shifts its position, moving it just far enough to expose a stretch of DNA previously hidden beneath its coils. The movement is gradual, unfolding over many small steps, and it does not loosen the surrounding nucleosomes unnecessarily. Only the region that needs attention is gently opened. Once the necessary enzymes have completed their work, the nucleosome can drift back into place, restoring the calm structure of the chromatin fiber. Another form of remodeling involves temporarily loosening the DNA's grip on the histone core. Histones hold the DNA through a combination of chemical attractions, but these bonds are not rigid. They respond to signals within the cell, particularly to small chemical tags that can be added or removed from the histone's tails. These tags, such as acetyl or methyl groups, act like quiet markers that instruct the chromatin whether to remain compact or loosen slightly. When a histone receives a signal to open, the DNA gently unwinds from the protein core, revealing a region of its sequence that may have been hidden for long periods. The process feels almost like lifting the edge of a page to read the text beneath. These histone modifications are among the softest forms of regulation in biology. They do not alter the DNA itself, nor do they permanently change the structure of the chromatin. Instead, they provide a reversible method of shifting accessibility, temporary openings that allow the cell to respond flexibly to its needs. When the moment passes, the tags can be removed, and the chromatin returns to its resting state. This dynamic environment allows the cell to maintain stability while also embracing change, balancing the need for protection with the need for access. Chromatin remodeling is especially important during DNA repair. When damage is detected, the chromatin around the affected region must quickly and gently open, allowing repair enzymes to reach the site. If a stretch of DNA is tightly wrapped, enzymes cannot access the damaged bases, no matter how skilled they may be. Chromatin remodelers respond by loosening the structure, sliding nucleosomes away from the damaged area, or introducing chemical tags that encourage the DNA to unwind slightly. This opening does not disturb the rest of the nucleus. It is precise, contained, and purposeful. Once the repair is complete, the chromatin restores itself with equal elegance. Histones reclaim their positions, nucleosomes slide back into alignment, and the DNA rewraps around its protein cores in smooth, even loops. The repaired region becomes indistinguishable from the surrounding chromatin, settled into the same gentle arrangement that characterizes the genome at rest. This return to order is as important as the initial opening, ensuring that the DNA remains protected and properly organized for future moments of reading or repair. The act of chromatin remodeling also reflects broader principles of biological organization. Just as the body maintains homeostasis through gentle adjustments, altering hormone levels, shifting nutrient flow, or signaling organs to respond to subtle changes, the nucleus maintains genomic balance through the soft adjustments of chromatin structure. Nothing in these processes is sudden or abrupt. They unfold in a calm cycle of opening, repairing, or reading, and closing again, much like the unrolling and re-rolling of a scroll. Chromatin remodelers work in harmony with the rest of the cell's repair machinery. Polymerases, ligases, helicases, and other enzymes rely on the openings provided by remodeling complexes, slipping into place only when the chromatin has been gently lifted. The interplay between these systems is beautifully coordinated. Remodelers do not operate alone. They respond to signals from the repair pathways, from the cell cycle checkpoints, and from the watchful surveillance proteins that detect changes in DNA structure. Every step is tied to a soft cascade of molecular communication. This coordination ensures that the chromatin remains both protective and responsive, offering stability without rigidity. The DNA, though wrapped and organized, is never locked away. It rests, accessible when needed and shielded when not, in a dynamic equilibrium that supports the entire life of the cell. As you imagine the chromatin in this peaceful environment, the long strands of DNA wrapped in their quiet spirals, you may sense the deep tranquility that underlies this process. Even though the genome contains billions of base pairs, each nucleosome acts like a soft anchor, each histone a gentle guardian, each remodeler a thoughtful guide who knows when to reveal and when to conceal. In this way, chromatin remodeling becomes more than a molecular mechanism. It becomes a symbol of the balance inherent in living systems. The genome is protected yet accessible, stable yet adaptable, resting yet ready. And through these gentle shifts, the DNA remains readable, repairable, and resilient, sustained by the quiet hands that open and close the shelves of the genetic library with unwavering care. 15. Mitochondrial DNA. Repair. Beyond. The nucleus. Deep within the quiet expanse of the cell, surrounded by the gentle currents of the cytoplasm, there exists another type of DNA, smaller, more compact, and carried within an organelle that hums with steady purpose. These are the mitochondria, often described as the cell's sources of energy, their interiors glowing with the soft chemical warmth that fuels life's daily rhythms. And within each mitochondrion, nestled amid folded membranes and calm biochemical cycles, rests a circular molecule known as mitochondrial DNA. It is quieter, simpler, and more ancient than the DNA in the nucleus, bearing a different history and requiring its own unique forms of care. This DNA, though modest in size, is essential for life, and like all genetic material, it experiences wear and change. Its repair processes, though less elaborate than those of the nucleus, unfold with a quiet resilience that reflects the delicate balance within these organelles. To imagine mitochondrial DNA, you might picture a tiny loop of genetic material drifting gently within the inner compartment of the mitochondrion. Unlike the nucleus, which houses long, carefully wrapped chromosomes, the mitochondrion holds only a handful of genes, just enough to guide certain crucial steps in energy production. These genes are surrounded by a warm glow of biochemical activity, for the mitochondria perform oxidative phosphorylation, a process that releases energy in the form of ATP. This process, essential for movement, thought, sensation, and the quiet hum of metabolism, produces small, reactive molecules as a natural byproduct. These molecules, often referred to as reactive oxygen species, float through the mitochondrial space like soft sparks. They are not inherently harmful, but over time they can nudge the DNA, gently altering bases or creating small disturbances in the circular strand. Because of this constant exposure, mitochondrial DNA encounters a steady rhythm of chemical wear. Yet the mitochondria do not experience this as a crisis. Instead, they have evolved repair mechanisms suited to their environment processes that are simpler and quieter than those in the nucleus, but no less dedicated to maintaining stability. These mechanisms rely on many of the same enzymes found in nuclear repair, yet tailored to the conditions within the organelle. They reflect a different kind of biological wisdom, one shaped by the fact that mitochondria trace their origins back to ancient bacteria that once lived freely in the world. Over billions of years, These ancestral bacteria became symbiotic partners within cells, passing on both their usefulness and their own specialized approaches to genomic care. One of the primary repair pathways in mitochondria resembles base excision repair, the gentle system used in the nucleus to correct small chemical changes in individual bases. When a base in mitochondrial DNA becomes altered perhaps by oxidation or deamination, small glycosylase enzymes move across the circular molecule, searching for these changes in the same flowing manner seen in nuclear repair. These glycosylases remove damaged bases, leaving tiny gaps that are then filled by mitochondrial DNA polymerase, often referred to as polymerase gamma. This polymerase carries out its work with quiet, deliberate precision, placing nucleotides into the vacant spaces and restoring the circular DNA to a continuous, unbroken structure. The process ends with the action of mitochondrial ligase, which seals the final bond, much as ligases do in the nucleus. Because mitochondrial DNA is packaged differently without the same extensive chromatin structure found in the nucleus, it remains more accessible to repair enzymes. There are no tightly wound histone cores to unwrap, no extensive remodeling complexes needed to reveal hidden bases. The DNA floats freely, its loops exposed within the matrix, making it easier for repair proteins to locate their targets. This openness reflects the organelle's bacterial ancestry, where DNA existed in a similarly uncluttered state. Yet this accessibility comes with a softness that requires constant watchfulness. The mitochondria rely on their repair enzymes to patrol and mend the DNA in an environment where damage arises regularly from energy production itself. Even with these repair systems, mitochondrial DNA accumulates changes over time. These changes are not viewed as failures but as natural aspects of the organelle's life, much like the gentle aging of tissues throughout the organism. With every cycle of respiration, every moment of metabolic activity, the mitochondria generate the energy needed for movement, thought, and the simple act of being, and with each cycle, their DNA experiences small shifts. The repair systems address many of these, yet some changes persist, becoming part of the quiet history carried within the organelle. These accumulated variations reflect the dynamic nature of life, where stability and change coexist in a gentle balance. Mitochondrial repair also differs in that the organelle often contains many copies of its DNA, dozens, sometimes hundreds, of identical loops drift within a single mitochondrion. This redundancy offers a kind of soft buffer against damage. If one copy becomes slightly worn or altered, other copies can compensate. The mitochondrion can even use intact copies to guide certain types of restoration, although it does not perform homologous recombination with the same elegance found in the nucleus. Instead, it relies on simpler comparisons and selective replication, allowing the most stable copies to persist over time. This quiet persistence creates an internal equilibrium, much like the balance maintained in large biological systems where multiple components share the same function. Another soft form of mitochondrial care emerges through turnover. Mitochondria are not static structures. They grow, divide, fuse with one another, and sometimes are gently dismantled when they become too worn. This cyclical process, known as mitochondrial dynamics, helps preserve overall health within the cell. When a mitochondrion becomes damaged beyond repair, signals guide it toward a gradual breakdown and recycling through a process called mitophagy. The cell does not treat this as loss but as renewal, creating space for healthier mitochondria to thrive. Through this process, mitochondrial DNA that carries significant damage can be quietly removed from the population, ensuring that the organelle community remains balanced and functional. The simplicity of mitochondrial repair reflects the environment in which the organelle operates. Unlike nuclear DNA, which is involved in countless forms of gene regulation and must be preserved with extraordinary precision, mitochondrial DNA guides only a small set of essential functions. These genes support the enzymes that allow the mitochondrion to produce ATP, participating in the elegant chain of reactions that convert nutrients into usable energy. Because their role is highly specialized, the repair systems focus on maintaining functionality rather than perfect fidelity. This focus underscores a softer truth. Biological systems care most about preserving harmony, not necessarily preserving identical sequences, and so within each cell, a quiet partnership unfolds. The nucleus tends large, detailed chromosomes wrapped in protective layers, while the mitochondria tend small circular molecules shaped by energy and ancient history. Both forms of DNA are essential, both require care, and both are maintained by systems that operate with gentle persistence. Yet each reflects a different aspect of life's story. The nucleus, with its sweeping instructions and deep generational continuity, and the mitochondrion, with its compact genes shaped by motion, heat, and chemistry. As you picture the mitochondrial DNA floating in its softly glowing space, touched occasionally by repair enzymes that glide across its surface, there is a sense of quiet resilience. Here, far from the structured world of the nucleus, life continues with a different kind of rhythm, simpler, older, and deeply connected to the energy that sustains every moment of being. The DNA within each mitochondrion continues its cycle of wear and restoration, supported by pathways that are humble yet steadfast, preserving the organelle's capacity to fuel the cell with gentle constancy. Through these soft mechanisms, the mitochondria and their DNA remain faithful partners in the ongoing work of life, contributing their energy, their history, and their quiet strength to the harmony of the cell. 16. Within the calm interior of every cell, where the DNA drifts in its gentle coil and the soft machinery of repair works tirelessly to maintain harmony, there exists a quiet truth that unfolds over time. Even the most patient processes gradually change. The enzymes that glide along the double helix, the remodelers that open chromatin with delicate care, the polymerases and ligases that restore broken regions all continue their work throughout life. Yet as the years pass, subtle shifts begin to appear. Movements that were once swift become slightly slower. Repairs that once occurred instantly take a moment longer. It is not a failure or a breakdown, but rather the natural softening that accompanies the passage of time. This gradual slowing is one of the most gentle manifestations of aging at the cellular level, a soft march forward that reflects the intimate relationship between life, time, and change. To imagine this process, picture the cell as a quiet room illuminated by a warm, diffused light. Inside this room, the DNA rests like an ancient scroll, its surface tended continuously by the soft-spoken helpers who maintain it. These caretakers never truly stop their work, but over the years, their rhythms shift. They arrive just a little less readily, or take slightly longer to make adjustments. Their abundance changes as well. Some repair proteins are produced in smaller amounts, some are regulated differently, and some respond to signals with a muted sensitivity. These shifts accumulate slowly, almost imperceptibly, creating a landscape where wear is not completely erased, but accommodated with patient acceptance. One of the most significant changes in aging cells is the gradual accumulation of small, unrepaired alterations in the DNA. These alterations are not catastrophic. They are more like faint marks on a well-read page, small reminders of moments when the repair systems were unable to act with their original speed. Every day, cells encounter countless tiny forms of DNA wear, from oxidative changes caused by normal metabolic activity, to soft distortions produced by sunlight, to minor replication slips. For much of life, the repair pathways address these imperfections with remarkable efficiency. But as the cell ages, some of these imperfections persist. The surveillance proteins may detect them more slowly, the chromatin may not open as readily, or the polymerases may move with slightly less precision. Over time, the accumulation of these small marks gently shapes the aging cell. The DNA remains functional, but certain regions become shrouded in subtle modifications, chemical tags on histones, changes in repair frequency, or structural shifts in chromatin that alter the accessibility of genes. These epigenetic changes are some of the quietest forms of cellular aging. They do not alter the DNA sequence itself, but they influence how easily the cell can read and repair its genetic material. Some genes become more tightly wound and less accessible, while others become more open than before. The cell continues to function within these changes, adapting to its altered landscape with the same patience that defines all living systems. Mitochondrial DNA also experiences its own gentle aging. As the mitochondria produce energy, they expose their circular DNA to a more reactive environment. And although they repair many of the small changes that occur, some persist. Over time, the mitochondrial population within a cell reflects these gradual shifts, carrying tiny variations that accumulate across the decades. Yet even here, the cell responds not with alarm, but with balance, retiring heavily worn mitochondria through mitophagy and allowing healthier ones to continue their steady work. It is a dance of renewal and release, quiet and continuous, shaped by the soft progression of time. Telomeres, the natural ends of chromosomes, offer another view into the aging process. These protective caps shorten slightly with each cell division, creating a calm form of biological counting. They do not fray abruptly but shorten gradually, strand by strand, until they reach a length that signals the cell to pause its divisions. This telomere shortening is often described as a marker of aging, yet within the cell, it feels more like a gentle indicator of time's passage, an internal clock that respects the need for balance, stability, and eventual rest. When telomeres become too short, the cell enters a state called senescence, a peaceful kind of retirement where it no longer divides but remains alive, contributing to the tissue environment through quiet metabolic activity and communication with neighboring cells. Senescence itself is not a harsh condition. It is a calm transition, a place where cells reduce their activities and settle into a slower rhythm. They no longer prepare for division, nor do they engage in complex replication processes. Instead, they remain present, maintaining their structure and interacting subtly with their surroundings. These senescent cells accumulate slowly within tissues, becoming part of the soft mosaic of aging. They carry with them the long history of wear, repair, and adaptation that shaped their lives. The slowing of DNA repair also affects how cells respond to external changes. Throughout youth, cells adjust to fluctuations in temperature, nutrient availability, and internal signaling with quick responsiveness. But with aging, these adjustments become more measured. Repair pathways may require more time to correct imperfections. Chromatin remodeling may shift more gradually. These changes do not signal dysfunction. Rather, they reflect a life lived through countless cycles of activity, each one leaving its soft imprint. Even the nucleus itself changes gently with age. The nuclear envelope becomes slightly less flexible, and the distribution of chromatin shifts into new patterns. These changes alter the flow of information within the cell, leading to differences in how genes are expressed. Some of these differences support continued cellular function, while others contribute to the broader tapestry of aging. Yet within the nucleus, the environment remains fundamentally calm. The DNA does not fray with panic. It adjusts, refolds, and continues its work. Throughout these changes, the cell demonstrates resilience. Aging is not a story of failure, but of adaptation. The gradual slowing of repair systems allows the cell to continue functioning even when some imperfections persist. Cells across tissues work together to maintain overall harmony within the organism, sharing signals, supporting their neighbors, and participating in processes that help rejuvenate or maintain tissue health. The cooperation among cells reflects the same unity observed in DNA repair, multiple systems acting together with quiet, steady purpose. And beyond the cellular level, the organism itself adapts to the gentle march of time. Metabolism shifts, tissues reorganize their balance of young and old cells, and organs adjust their rhythms. These changes, like those within the cell, unfold slowly. They do not disrupt life suddenly, but instead create a new equilibrium, shaped by a lifetime of interactions between biology and the environment. As you imagine the aging cell, with its softly worn DNA and its still dedicated repair machinery, a sense of calm emerges. The cell continues its work with patience, tending to its genetic material as best it can, comforting itself through its many pathways of care. The marks of time become part of its identity, woven gently into the loops of chromatin and the coils of mitochondrial DNA. Through these soft transformations, the cell remains a living testament to the balance between preservation and change. In this way, cellular aging becomes not a story of loss, but a quiet reflection of life's enduring processes. The DNA, though touched by time, still guides the cell with its familiar instructions. The repair systems, though slower, still offer their gentle support, and the cell, ever resilient, continues to move forward one soft moment after another, carrying the history of its experiences with grace. 17. DNA repair and evolution, preserving life through change. Within the gentle interior of the cell, where the DNA coils in quiet symmetry and repair systems move with calm precision, a subtle and profound truth unfolds. Life is shaped not only by the preservation of the genetic code, but also by its capacity for change. DNA repair systems work tirelessly to correct disturbances, restoring harmony to the double helix whenever possible. Yet even in their diligence, these systems allow small variations, tiny shifts in the sequence that slip softly through the filters of correction. These changes, though often unnoticed within the life of a single cell, become part of a much larger story, the story of evolution, the patient shaping of life across countless generations. It is here, in the balance between precision and variation, that the quiet interplay between repair and change reveals its deepest significance. To imagine this interplay, you might picture the DNA not only as a blueprint for a single organism, but also as a thread woven into a tapestry that stretches across time. Every slight adjustment in the sequence, every alteration that persists through replication, becomes a potential new pattern in the fabric of life. Yet these patterns do not appear all at once. They emerge through slow, gentle processes, base by base, generation by generation, guided by the soft rhythms of repair, replication, and natural variation. The cell's repair systems act as careful guardians, ensuring that the DNA remains functional and stable, but they also allow room for subtle modifications that do not disrupt the overall harmony of the organism. It is within this delicate balance that evolution quietly unfolds. DNA repair pathways are astonishingly accurate, particularly those such as homologous recombination and mismatch repair. They recognize disturbances with remarkable sensitivity, correcting mismatches and restoring double-strand breaks with fidelity. Yet even these systems are not absolute. A mismatch may occasionally evade detection, becoming a permanent part of the DNA sequence after replication. A double-strand break repaired through non-homologous end joining may result in the addition or deletion of a small number of nucleotides. A base, altered by chemical wear, may be replaced with a slightly different nucleotide than before. These changes are not errors in a negative sense, they are natural variations that arise from the interplay between the cell's desire for stability and the realities of its environment. Over long periods, these small variations accumulate. The genetic code is filled with redundancies, multiple sequences that encode the same amino acid, allowing many changes to pass quietly through. Others may influence the shape or behavior of a protein ever so slightly, altering the way it folds or interacts with other molecules. These changes may be harmless, or they may subtly influence the efficiency of a biochemical pathway, the sensitivity of a receptor, or the timing of a developmental process. Still others may affect gene regulation rather than the structure of proteins, modifying how often a gene is expressed, or in what context, or to what extent. In the vast majority of cases, these gentle shifts remain part of the organism's genetic background without causing harm. They may even persist unchanged across generations, carried within the quiet architecture of the genome. But sometimes, a change becomes meaningful, offering a small benefit to the organism in its environment. It might make a protein more stable in a slightly warmer climate, or allow an enzyme to process nutrients more efficiently, or influence behavioral patterns that improve the chances of survival. When these advantages arise, natural selection, the calm but persistent force described in the principles of evolution, favors individuals who carry the beneficial variation. Over generations, the variation becomes more common, gradually shaping the traits of the species. This process is slow, subtle, and deeply intertwined with the cell's ability to repair and preserve its DNA. Without repair, the genome would accumulate changes too rapidly, causing instability that would overwhelm the organism. Without variation, the genome would remain static, unable to adapt to changing environments or new conditions. It is the gentle balance of these forces that allows life to evolve. Repair protects the essential functions of the cell, ensuring that the machinery of life continues to operate with precision. Variation introduces opportunities for novelty, creativity, and adaptation, allowing life to explore new possibilities across vast stretches of time. Mitochondrial DNA offers another perspective on this balance. Because mitochondria are exposed to reactive byproducts of energy production, their DNA accumulates variations more quickly than nuclear DNA. These variations, passed down through the maternal line, create patterns that reflect ancient migrations, adaptations to environment, and the quiet history of human populations. Over thousands of years, these mitochondrial changes provide clues to the paths our ancestors traveled, the climates they encountered, and the ways they adapted. They are gentle signatures written in the soft loops of mitochondrial DNA, preserved through the interplay of wear, repair, and inheritance. Similarly, the chromatin structure surrounding nuclear DNA plays a role in evolution by influencing which regions are more accessible to change. DNA wrapped tightly around histones is less likely to undergo mutation, simply because it is more protected. Regions that are loosely wrapped or frequently opened for transcription are more exposed, more dynamic, and more prone to subtle shifts. Over time, these patterns of accessibility shape the landscape of evolution, creating areas where variation occurs more readily and others where stability is preserved. Another important aspect of this interplay is recombination during meiosis, the process by which reproductive cells are formed. Unlike the repair-focused homologous recombination used during DNA repair, meiotic recombination is designed to create variation. Here, whole segments of DNA are exchanged between paired chromosomes in a slow, graceful dance that mixes genetic material from both parents. This process ensures that each offspring receives a unique combination of alleles, contributing to the diversity that fuels natural selection. Meiotic recombination is one of the most intentional forms of variation in biology, yet even it unfolds with the same quiet order that characterizes the rest of the cell's molecular processes. Across generations, the variations that persist reflect the environments in which organisms live. In colder regions, genes that influence metabolism may shift. In regions with abundant sunlight, protective pigments may become more common. In environments with scarce resources, traits that improve efficiency or reduce energy consumption may proliferate. These changes arise slowly, guided by the interplay between the gentle creation of variation and the quiet consistency of repair. Evolution does not rush. It moves at the pace of life itself, soft, steady, and deeply responsive. As you imagine this process unfolding across countless lifetimes, the scale becomes almost soothing. Individual cells continue their daily routines, repairing, replicating, resting, while the genome they protect carries a history that stretches far beyond their own existence. Each small variation is like a whisper, a quiet note in the symphony of life, contributing to the gradual shaping of species, ecosystems, and the broader patterns of biodiversity on Earth. In this sense, DNA repair is not merely a mechanism of preservation, it is a guardian of potential. By correcting the majority of disturbances while allowing a few gentle shifts to persist, the repair systems ensure both stability and adaptability. They protect the organism while enabling evolution. They maintain the present while supporting the future. Thus, the cell's quiet repair pathways become part of a much larger rhythm, one that transcends the boundaries of a single lifetime. They participate in the soft continuity of life, preserving the integrity of the genome while allowing the gentle changes that shape the unfolding story of Earth's living beings. Through this delicate balance, life remains both resilient and ever-changing, carrying its history forward in the calm and enduring spirals of DNA. 18. Repair, in the context of the whole organism. When we imagine DNA repair, it is easy to picture these processes occurring in quiet isolation within a single cell, unfolding in the gentle stillness of the nucleus. Yet each cell is part of a much larger tapestry, an interconnected system where countless cells work together to sustain the life of the organism. The repair of DNA, though it happens on a microscopic scale, influences the health, stability, and resilience of the entire body. In turn, the body's broader systems, circulatory, endocrine, immune, nervous, create conditions that support or influence the quality of DNA repair. When viewed in this wider context, DNA repair becomes not merely a cellular act, but a quiet conversation between all levels of biological organization, from molecules to tissues to the whole living being. To imagine this relationship, think of the organism as a calm and expansive environment, where each cell resembles a small house within a vast, peaceful village. Each house contains its own routines, its own caretakers, and its own maintenance systems. The DNA repair pathways are like the skilled artisans who care for the structure of each house, fixing small cracks and reinforcing its foundation. Yet the health of each house is also influenced by the surroundings, the climate, the flow of resources, and the harmony of the community. In the body, this means that DNA repair does not occur in isolation. It is supported by the flow of nutrients, the balance of hormones, the efficiency of waste removal, and the gentle orchestration of homeostasis that keeps the internal environment stable. Homeostasis plays a particularly important role. The body maintains narrow ranges for many conditions: temperature, pH, glucose levels, and oxygen availability, ensuring that the interior remains calm and predictable. These stable conditions are essential for DNA repair enzymes to function optimally. For example, polymerases and ligases depend on the availability of certain ions and cofactors, which the body carefully regulates through digestive absorption, circulatory distribution, and kidney filtration. The gentle regulation of calcium levels, the maintenance of hydration, and the steady supply of nutrients all support the internal environment in which DNA repair enzymes can move with precision. Even small shifts in homeostasis can subtly affect repair efficiency, reminding us that the organism's quiet internal balance is inseparable from the health of its cells. The circulatory system also contributes to this harmony, delivering oxygen and nutrients to every cell. Oxygen enables energy production within mitochondria, giving repair enzymes the fuel needed to perform their duties. Blood flow brings glucose, amino acids, vitamins, and minerals, all essential for synthesizing new nucleotides and repair proteins. When circulation is smooth and uninterrupted, cells receive everything they need for efficient repair. Conversely, regions with limited blood flow may experience slower repair simply because fewer resources reach the cells. This relationship reveals how deeply the act of DNA maintenance depends on the body's ability to sustain each cell with gentle, continuous support. The endocrine system, with its soft waves of hormonal signaling, also plays a role in DNA repair. Hormones act as quiet messengers, traveling through the bloodstream and influencing cellular activities in subtle ways. Stress hormones can temporarily shift the cell's priorities, guiding energy toward immediate responses and away from long-term maintenance. In contrast, hormones that regulate growth, metabolism, and reproduction often stimulate cells to prepare their DNA, replicate it, or repair small irregularities before division. These hormonal cues help the body coordinate cellular behavior, ensuring that tissues grow, adapt, and renew themselves in a controlled and harmonious manner. The immune system contributes in yet another way. Immune cells constantly monitor tissues, identifying and removing cells that carry significant damage cells, whose DNA may have experienced too much wear, or whose repair systems have slowed beyond recovery. This removal is not aggressive but necessary, preventing damaged cells from disturbing the broader community. In this sense, immune surveillance forms an outer layer of repair. By clearing damaged cells and supporting tissue renewal, the immune system helps maintain the overall integrity of the organism. It interacts with DNA repair indirectly but meaningfully, shaping the conditions in which cells can thrive. Even the nervous system quietly influences the environment for DNA repair. During restful states, repair often becomes more active. Sleep, in particular, provides the body with a period of reduced external stimulation, allowing cells to focus on internal tasks, including DNA repair. Within the gentle stillness of sleep, repair proteins can move with fewer interruptions, and damaged cells have the opportunity to restore themselves more effectively. This interplay between consciousness, physiology, and DNA repair illustrates how deeply connected these seemingly separate layers of biology truly are. Tissues themselves shape the context for repair. Different tissues divide at different rates. Skin cells, intestinal cells, and blood cells divide frequently, requiring constant repair to preserve accuracy during replication. In contrast, neurons and muscle cells divide rarely, relying more on precise maintenance and long-term protection of their DNA. The structural and functional diversity of tissues means that DNA repair is not uniform throughout the body. It adapts to the needs and rhythms of each cellular community. Even within a single organ, some cells may carry more metabolic responsibility, more mechanical stress, or more exposure to environmental influences, leading to different patterns of wear and repair. As cells age and accumulate small marks from a lifetime of repair and replication, the tissues they form also change. The organism gradually adjusts to these shifts, finding new patterns of balance. This mosaic of aging cells influences the overall environment, shaping how effectively the remaining cells can repair their DNA. The process becomes a gentle feedback loop. The organism supports the cells, and the cells maintain the DNA that sustains the organism. Together, they move through time with quiet resilience. The organism also provides a larger context for evolutionary variation. While DNA repair prevents harmful changes, the organism's ability to reproduce and pass on its genetic material ensures that beneficial variations can accumulate across generations. In this way, the health of individual cells contributes to the stability of the species, while the species environment influences which variations persist. DNA repair thus becomes a bridge between molecular precision and evolutionary possibility, carried within the living bodies of organisms that thrive in diverse habitats. The environment outside the organism further shapes the conditions for repair. Sunlight, temperature, nutrients, and natural chemicals all influence cellular wear. The organism responds by adjusting behavior, seeking shade, regulating body temperature, consuming nutrients, or storing energy to protect its cells. These behaviors, in turn, influence the DNA repair processes within each cell. The interplay between organism and environment is a soft yet powerful force. As you picture this multi-layered relationship, the act of DNA repair begins to feel like one of the many threads in the fabric of life, a thread that runs from the smallest molecular interactions to the full complexity of an entire organism. Every repair, no matter how small, contributes to the stability of tissues, organs, and systems. Every moment of homeostasis supports the repair pathways within cells. Every breath, every heartbeat, every gentle wave of blood flow helps create the conditions for DNA to remain whole. In this way, the quiet work of repair becomes part of the organism's deeper harmony. It unfolds within cells, but its effects radiate outward, shaping health, growth, adaptation, and resilience. Through this soft network of support, the DNA remains a steady guide, carrying the instructions for life while being cared for by the systems it helps sustain. The cell, the tissue, the organ, and the entire organism move together in a shared rhythm, preserving the balance that allows life to flourish from its smallest molecules to its grandest forms. 19. Environmental influences. Within the quiet interior of every cell, the DNA rests in its soft, looping form, tended continuously by watchful enzymes and protected by layers of chromatin. Yet the cell does not exist in isolation. It lives within the body, and the body lives within the world, a world filled with light, temperature, nutrients, rhythms, and countless subtle influences. These environmental factors do not often strike the DNA with force. More commonly, they act as gentle pressures, shaping the cell's long-term needs for repair, renewal, and adaptation. The interactions between the environment and the genome unfold gradually, like soft waves brushing against the shore, leaving patterns that the DNA repair systems learn to interpret and respond to over time. To imagine this relationship, think of the organism as a serene landscape surrounded by the natural world. The body breathes in air, absorbs light, nourishes itself from food, and responds to the rhythms of day and night. These environmental elements move through the tissues and cells, carrying signals, energies, and molecules that touch the DNA indirectly, encouraging the cell to protect itself, repair itself, or adapt in quiet and enduring ways. Rather than dramatic damage, most environmental influences create small nudges, tiny chemical shifts, faint molecular impressions, or slow accumulations of mild wear. And just as the environment shapes the organism responds by activating repair pathways that preserve the gentle continuity of life. Among the gentlest and most pervasive of these influences is sunlight. When light touches the skin, its energy enters the cells and interacts with the DNA. Ultraviolet rays, though soft compared to more intense forms of radiation, can cause thymine bases to gently bond with one another, creating tiny dimers that introduce slight ripples in the helix. These distortions do not shatter the DNA. They simply reshape it in subtle ways that the nucleotide excision repair pathway is well designed to correct. The cell opens a small window in the chromatin, removes the affected region, and restores the sequence with calm precision. Over a lifetime, this quiet process repeats itself countless times, guided by the gentle balance between sunlight's warmth and the cell's protective care. Temperature is another environmental factor that affects the genome indirectly. The body maintains its internal temperature through homeostasis, ensuring that cells remain within a calm and comfortable range. Yet, slight changes in temperature, those caused by seasons, time of day, or physical activity, can influence the fluidity of cellular membranes, the speed of molecular movements, and the efficiency of repair enzymes. A warmer environment may allow certain chemical reactions to occur more quickly, while cooler temperatures may slow them down. The cell adapts to these shifts with patience, adjusting enzyme activity, rearranging chromatin when necessary, and ensuring that DNA repair continues smoothly regardless of external fluctuations. The nutrients the body absorbs from food also shape the DNA's environment. Vitamins such as folate and B12 support the synthesis of nucleotides, helping the cell create the building blocks needed for repair. Minerals like zinc and magnesium act as quiet cofactors, stabilizing the structure of repair enzymes and guiding the formation of bonds. Antioxidants found in fruits and vegetables help neutralize the reactive molecules produced by normal metabolism, reducing the chemical wear on DNA. Even the simplest meal becomes part of the environmental conversation between the world and the genome, providing the resources needed to maintain the soft resilience of the nucleus. At the same time, the metabolic processes that sustain life create their own gentle pressures on DNA. As mitochondria produce energy, they generate reactive oxygen species that drift through the cell like tiny sparks. These molecules can occasionally interact with DNA bases, creating small chemical shifts that repair systems must correct. The process is natural, continuous, and woven into the rhythm of the cell's daily life. The environment inside the organism, shaped by breathing, movement, digestion, and rest, interacts constantly with the genome, creating a slow and steady pattern of wear that the cell has evolved to manage gracefully. Water, perhaps the most essential environmental element, also influences DNA indirectly. Hydration affects the fluidity of the cytoplasm, the shape of proteins, and the ability of enzymes to move along the double helix. A well-hydrated cell experiences a smoother internal environment, allowing repair proteins to glide more easily through the nucleus. Even dehydration, when mild and temporary, responds to the body's balancing systems, prompting quiet adjustments that ensure the DNA remains protected. This interplay reflects the delicate dependence of cellular processes on the water that permeates all living structures. Another gentle environmental influence arises from the presence of natural chemicals in the world. Plants, soil, air, and water all contain organic compounds that may enter the body in small amounts. Some of these compounds interact peacefully with the cell, while others cause mild chemical modifications to DNA bases. These interactions occur gradually, often as part of long-term environmental exposure. The cell responds by activating glycosylases that remove altered bases, polymerases that rebuild the sequence, and ligases that restore continuity. Rather than overwhelming the cell, these chemical interactions become part of the ongoing conversation between the organism and its surroundings. Behavioral rhythms also shape DNA repair in quiet ways. Exposure to natural light during the day and darkness at night helps regulate circadian rhythms, which influence the timing of cellular repair. During sleep, when the body settles into a restful, restorative state, repair systems often become more active, chromatin opens more readily, enzymes move more freely, and the cell takes advantage of the reduced metabolic demands to focus on internal maintenance. In this sense, the environmental rhythm of day and night becomes a soft guide, encouraging the cell to perform repair during periods of calm and restoration. Even the broader climate contributes to the gentle shaping of genomic maintenance. Humidity influences the hydration of tissues, while atmospheric oxygen levels guide mitochondrial activity. Seasonal changes affect hormone levels, nutritional availability, and patterns of movement, all of which indirectly shape DNA stability. Life in colder regions may encourage stronger protective pathways against slight changes in temperature, while life in sunnier regions may favor efficient correction of light-induced distortions. The environment becomes an external partner in the cell's internal balance, offering cues that the genome interprets across generations. For the organism as a whole, environmental influences create a canvas on which DNA repair paints its delicate patterns. Cells within the lungs experience different conditions from those in the liver, and skin cells touching the outside world more directly experience a different kind of wear than cells deep within the brain. Each environment shapes the needs of its cells, and each cell adapts its repair pathways accordingly. This diversity reflects the deep interconnectedness between the organism's multiple environments: external, internal, chemical, and rhythmic, and the internal maintenance processes that preserve the genome. Over long periods, environmental influences also contribute to evolutionary change. Regions that expose organisms to particular patterns of sunlight, climate, or nutrition gently shape the genomic variations that persist. The DNA repair systems do not prevent these shifts. Instead, they manage them, ensuring that only small, tolerable changes accumulate. This interplay between environmental pressure and molecular resilience allows life to adapt gradually, creating diverse forms and behaviors across ecosystems. As you imagine the DNA resting within the quiet interior of a cell, surrounded by the soft influences of environment and the steady guardianship of repair enzymes, the entire process begins to feel deeply harmonious. The world touches the genome gently, never in isolation, but always through layers of protection, adaptation, and response. The cell listens to these cues, adjusting its repair pathways with patience. The organism interprets the environment, shaping the conditions in which cells can flourish. And together, the genome and the world form a continuous dialogue, one made not of force, but of soft, enduring contact. Through this ongoing conversation, the DNA remains both stable and adaptable, protected by the quiet intelligence of repair systems that understand the language of the environment. And the organism, shaped by these subtle interactions, continues to thrive in harmony with the world that surrounds it. 20. The calm continuity of life. A gentle closing reflection. As we come to the final part of this quiet journey through the world of DNA repair, it becomes clear that the processes unfolding within each cell are far more than molecular routines. They are gentle acts of preservation, patient, rhythmic, and continuous, woven into every moment of the organism's life. From the soft removal of a damaged base to the careful sealing of a newly restored strand, the cell performs its duties not with urgency or force, but with a kind of serene devotion. The DNA, in turn, accepts this care with quiet resilience, holding the record of the past while guiding the possibilities of the future. Together, they create a harmony that underlies all living systems, sustaining life through balance, patience, and enduring continuity. If we step back to view this intricate system from a broader perspective, the calm logic of it becomes even more soothing. DNA is, at its essence, a vast library of instructions written in simple language, a sequence of bases arranged in gentle patterns that have persisted across millions of years. It does not rush. It does not demand attention. It simply rests in its spiraling form, waiting to be read, copied, or repaired. The repair systems that tend to it behave in the same quiet manner, gliding along the helix like peaceful custodians who move through dimly lit aisles of an ancient archive. Their actions preserve clarity, continuity, and order within the vast expanse of biological information. Throughout this journey, we have seen how various forms of DNA repair unfold with delicate precision. Base excision repair removes altered nucleotides one at a time, like a careful hand lifting a single grain of sand from a larger mosaic. Nucleotide excision repair opens wider windows to remove distorted patches. Mismatch repair corrects the small slips that occur during replication, restoring the smooth pairing of the bases. And when both strands of the helix experience an interruption, the cell responds through homologous recombination or non-homologous end joining, each pathway applying a different level of accuracy depending on the needs of the moment. None of these processes operate in isolation. Instead, they are part of an interconnected web of care that moves in harmony with the cell's cycle, its environment, and its deeper biological rhythms. Beyond the nucleus, we found that mitochondria carry their own compact DNA, shaped by ancient evolutionary history and cared for through specialized forms of repair. These circular molecules exist in a warm space filled with energy, gently exposed to the byproducts of metabolism. Their wear reflects the life and movement of the organism, while their repair reflects resilience. Through cycles of renewal and quiet recycling, the mitochondria adapt and persist, contributing their essential role in fueling the cell. We also saw that repair slows as the cell ages, not as a failure but as a natural shift in rhythm. The DNA accumulates small marks, chromatin reshapes itself, and repair systems adjust their pace. The cell enters new states of balance, finding harmony even as it changes. Cellular aging becomes a reflection of the organism's long journey, gentle, gradual, and deeply intertwined with the passage of time. Evolution, too, plays a quiet role in this story. These variations, soft and subtle, form the raw material from which species gradually adapt. Evolution is not a turbulent process. It is the slow unfolding of possibilities, guided by both preservation and change, generation after generation. And across all of these layers, the environment creates gentle pressures that shape the cell's needs for repair. Sunlight, temperature, nutrients, and natural rhythms all play quiet roles in shaping the genome's experience of the world. The organism responds through adjustments in metabolism, behavior, and cellular activity, ensuring that DNA repair can continue in a calm and balanced manner. When we consider all of these elements together, what emerges is a picture of life as a continuous interplay between care and adaptation. DNA repair is not merely a response to damage, it is a fundamental part of living, a daily expression of the cell's desire to remain whole. It reflects the elegance of biological systems, where precision and flexibility coexist, where change is allowed, but not left unchecked, where every moment is an opportunity to maintain harmony. This harmony extends beyond molecules and cells to tissues, organs, and the organism as a whole. The circulatory system delivers nutrients to support repair enzymes. The endocrine system provides quiet hormonal cues that shape cellular behavior. The immune system gently removes cells that can no longer maintain their DNA. The nervous system orchestrates rhythms of activity and rest that influence the timing of repair. All of these systems together form a softly coordinated environment in which DNA can be maintained with remarkable fidelity. And yet, despite the complexity of these mechanisms, the overall experience remains peaceful. The DNA is not overwhelmed by the world. It is supported by networks of care, evolved over billions of years. Each repair pathway, each enzyme, each chromatin shift contributes to the quiet miracle of continuity. Life moves forward because the genome is held in this web of gentle interactions. As we bring this reflection to a close, imagine the DNA once more in its serene spiral, drifting in the calm interior of the nucleus. The repair enzymes, the chromatin remodelers, the mitochondrial caretakers, each one continues its work, softly and steadily. The cell breathes in its own rhythm. The body moves through its cycles of waking and rest, and the environment beyond the organism offers its steady presence through seasons, light, and time. Together, these layers form the continuity of life, not through dramatic moments, but through quiet ones, not through sudden transformation, but through steady care. And this continuity is what allows every organism to grow, adapt, remember, and gently pass on its legacy to the next generation. In the end, DNA repair is not merely a defense system, it is a language of patience, a reminder that life persists not through strength alone, but through resilience expressed in the smallest, calmest moments. As long as the DNA is cared for, the patterns of life can continue, flowing softly from cell to cell, from organism to organism, extending through time like a peaceful and unbroken thread. And so we leave the double helix where it resides best, settled, supported, and serenely tended, carrying within its structure the quiet continuity of living things. In Stardy,